Hello, everyone. Thank you for joining us today in our Analytical Solutions for High-Dimensional Spectral Flow Cytometry Data. My name is Janelle Shook. I work at Cytek Biosciences, and I will be your facilitator for today. This event was actually an idea that we had for our user group meetings this year, but alas, since times have changed and in-person events aren't quite feasible at the moment, we adapted and we're going virtual, and all of our software partners were still very excited to be able to interact with everyone. Granted, it'd be nice to see everyone's faces and talk to you in person, but we'll do what we can with the question and answer window that we have available to us today. So to get you familiar with that webinar controller, if you have any questions that come up while any of the presenters are talking, go ahead and type it into the question box, and we will answer them at the end of each session, and also we'll have some time at the very end today to take any questions we didn't get to. The schedule for today will have 5 different presenters speaking from various software analysis companies, and they'll all be working through the same high quality 33 color data set that we Cytek provided for them to demo for you. And partway through, around 10:45 Pacific time, we'll take a 15-minute break so everyone can do their bio break and grab some water and be ready for the afternoon sessions. Now, to kick things off, I want to invite you all to participate in a poll so we can get an idea for who is all with us today. Let me get that kick-started. Okay, poll should have appeared on your screen. We'd like to know how many markers you typically include in your panels, and I'll give you all a few seconds to select an answer. Okay, looks like most everyone has submitted something, so I'm going to go ahead and close the poll and share the results with you. Seems like most people are operating in the 11 to 15 panel size range, and quite a few that are working on building up into the 20 and 30 plus space. That's great to know. Thank you for participating in that. Let me get that out of the way and continue on. I'd like to introduce our first speaker for today. Her name is Diana. She has a Ph.D. in immunology from Texas A&M University and a postdoc degree from Baylor College of Medicine. She is a cytometry specialist and has 20 years of experience in cellular evaluations for a variety of applications, including infectious disease and cancer. She was also an ISAC SRL emerging leader and worked as a senior scientist for the Medicine Nobel laureate James Allison, characterizing cytometric immune signatures in patients treated with cancer immunotherapy at MD Anderson Cancer Center. She's currently working at Cytek as one of our technical application specialists. So without further ado, I'm going to give control over to Diana so she can give her presentation. Here you go, Diana.
Thank you so much, Janelle, for the kind introduction, and also thank you everyone for attending this cool initiative. Current innovations in reagents and instrumentation allow for the measurement of unprecedented number of simultaneous parameters for thousands of individual cells in each sample analyzed. And there is a clear need for robust data analysis tools to explore those data sets and understand their complexity. But before you even think about the different analytical solutions, you need to make sure you have rock solid data that is trustable and reproducible, and do some work ahead to catch if there were any issues with sample preparation, staining, instrument performance, or any batch effects that need to be taken into account. Today, I will review the steps that you need to do before jumping into any of the options for high-dimensional analysis that we will see today. I will cover 3 topics. First, a brief introduction about our technology and about the data set that is going to be discussed today. Second, I will describe the guidelines on how to generate high-quality data, especially how to assess data quality. And last, some background slides on computational analysis before we hear more from our invited speakers. High quality data means that all your populations are easily resolved, especially rare subsets or markers with dim expressions. You want to make sure the photons emitted by fluorochromes, fluorescent proteins, or dyes are efficiently captured and resolved from noise or background on your cytometer. The Cytek Aurora instrument definitely provides very well-resolved high-dimensional data using the same operational principles as any other flow cytometer with some optical and fluidics improvements. In our system, that high resolution comes from the unique optical design. We use avalanche photodiodes, or APDs, as detectors of light. APDs have low electronic noise, have high quantum efficiency, and that translates into your negatives being low and tight and your positives being bright. In terms of multiplexing, the system is very flexible. With a single optical configuration, you can accommodate multiple fluorochromes. You can also extract autofluorescence or remove any light coming from unstained cells that can compromise the resolution of dim markers. Many other great features, live and mixing, more fluorochrome choices, volumetric counting. We provide you with Cytek assay settings, a set of optimized gains that are going to give you great resolution for multiple applications. And in addition, Aurora owners have access to panel and experimental design support and troubleshooting assistance from our team of scientists and engineers. Well, fluorochromes typically are excited by more than one laser and emit over a range of wavelengths. And Cytek has a different way to capture that information. The data set that we will discuss today was generated in a 5-laser unit. So after lasers excite the fluorochromes, we captured the entire emission across multiple APD modules, slicing the wavelength range into pieces, and then combining that information, giving you what we call a full spectrum signature, all the light that is emitting by that fluorochrome across multiple detectors based on the intensity of the signal, rather than identifying only the peak of emission. Each fluor has a unique spectral signature that identifies them, and if the signatures are unique enough, even for highly overlapping dyes, they can be used within the same panel. This technology can be used for multiple applications, including immunological studies. And now that we can build larger panels, we can expand the immunophenotyping and the understanding of the complexity of immune response. This particular panel explores human hematopoietic cells. We use multiple lineage markers to define the lymphoid and myeloid compartments and their different subsets, including lymphocytes, T cells, B cells, NK cells, dendritic cells. And then we use functional markers to define memory state as well as activation and exhaustion. This is the fluor selection for this panel. An important thing that is not just putting fluorochromes together, it's making sure that when you do that, you don't sacrifice resolution. For this panel, we identified 33 dyes that are all unique, with a mixture of brightness assigned based on antigen density and co-expression, with a manageable spread between fluorochromes, but especially making sure that all your populations have great resolution. This is going to give you a general idea of the multiple subsets that can be identified with this panel, using the traditional sequential manual gating strategy, where you inspect the plots in 2 dimensions at a time to identify the main populations of interest. But the slide also reveals that for this highly complex panel, manual gating has limitations. It's time-consuming, it's limited to the investigator knowledge, it's subjective, and it can be error-prone even in the hands of experts. And also a critical point is that you might be missing the presence of other populations, since we tend to focus gating only on cell subsets that we know or that are relevant for our study. So the goal for this workshop is to show you examples of computational tools that are commercially available to visualize this high-dimensional data set in a more explorative way. The first steps to generate high-quality data were already covered in our previous webinar by Dr. Maria James, and here's the link in case you want to learn more about. We have multiple tools available to build your theoretical panel in a much more informed way. But remember that it's critical not only panel design, but you validate the panel, that you optimize the controls you will use for unmixing, and also everything related to the assay, sample preparation, staining protocol, including things like antibody titration and buffer and washing conditions. After your panel has been validated, you want to make sure every time you use the panel, the data obtained is trustable. So these are 6 steps to assess data quality. First, you want to check your instrument, the settings used for acquisition, check your controls, how well those controls are going to go in terms of the unmixing. Make sure you remove any events that are not meaningful and that you evaluate the staining quality in your panel. So we'll review one by one. Number 1, check your instrument performance. Run daily QC and review the report. QC passing alone is a good indication that your instrument is performing well. So beats will be adjusted, we will adjust gains based on the beats intensity for all the detectors. Laser delays and area scaling factors will be calculated automatically. So look at the settings for your QC and the settings for your samples. If you're using Cytek assay settings, that should be similar. We also have an added bonus, the Levy-Jennings tracking, for customers to visualize the instrument performance across time and find if there's any issues. Step number 2, check your instrument settings, the ones that you use during sample acquisition. Starting with scatter settings, you should adjust your forward scatter and side scatter gains to have your population on scale. Also optimize thresholds so you don't record events that are going to be useful. Optimize forward or area scaling factors, especially for large cells, and check the stability of the signal across time. Remember that if you use the Cytek assay settings, it's so easy to create high quality data because the gains on Cytek assay settings for each detector have been optimized to give you great resolution, accurate signatures, and also low breadth. Part of number 2 is to make sure the settings are consistent across all the samples, and these are the 5 donors that we will discuss today. And not only the gain numbers, but also the type of signal, area, height, or width, referring to the geometry of the electronic pulse out of each single detector. Step number 3, check the quality of your reference controls. These are the single color controls that we use to calculate unmixing, or the contribution of each fluor in the panel to your multicolor sample. So in order to have good unmixed results, we need to feed the algorithm with optimal controls. You first need an unstained tube with a clean background that matches your fully stained sample in terms of same type of cells, same staining protocol, and same stimulation, if any. Reference controls. You need to check the negatives and the positive peaks for that control. Your positive should have the expected signature that is accurate, that is unique, that is bright, and also within the scale. Signature plots are also very powerful, not only to identify the signature, but to realize if there were any issues with fluorochrome mismatch, contamination, tandem degradation, or carryover. For the negative, you want to make sure it has a clean background with no contamination or carryover as well. Beyond that visual inspection of the signatures in your reference controls, there are other ways to check their quality using tools in the software. And the metrics that we have described before, like similarity index and complexity index, that are not only useful for panel design, but will help ensure the correct dye has been recorded. We have benchmark controls. Those can be saved in the Aurora library to then compare the emission with the acquired reference control using similarity index, which is a metric that tells us if 2 fluors have similar signatures with a range from 0 to 1, where 1 is identical emission profile. And this is important to capture reagent failures or any errors in staining or acquisition. If the spectral profile is not similar to the benchmark, then the similarity index will be low, close to 0, and the control will be flagged and you can decide to replace it. In terms of complexity index, it evaluates the overall similarity for all the fluors within the panel. The lower the complexity index on the bottom, the higher the chance of obtaining optimal results with a given combination of fluors. But it's also a great tool to make sure your controls are working well based on the expected similarity metrics. Step number 4, clean up your data. You need to remove events that should not be included in the analysis because they are technical artifacts or information not meaningful. Technical artifacts during acquisition that can impact your data quality. For example, bubbles or clogs can cause signal fluctuation. So you can use time gating to remove any areas with discontinuity, with low density events, or with large spikes. And also, to prepare the data for analytical profiling, you can gate out events such as cell debris, cell doublets or aggregates, dead cells, or even antibody aggregates if present. So you need to make sure that you remove any events that are not going to be meaningful. Also, if needed, you can remove cells that are not of your interest. Let's say you want to focus your analysis only on CD45 positive cells or only on the T cell compartment. Step number 5, check your unmixing accuracy. So during unmixing, what we do is we decompose the multicolor sample into multiple dyes relative to the reference controls. And an important step in quality control is to identify if there are any errors or issues with your unmixing. The best way to figure out if your unmixing is accurate is look at your sample. This is an example with single stain cells plotting all possible fluor combinations. In this case, BUV 395 versus all the other fluors in the panel. And what you need to look for is the alignment between the intensity of fluorescence for your positive and your negative in the y-axis, if it's a single color stain. And if it's a multicolor sample, you would look for that alignment on both axes, negative and positive, negative and positive. But in this case, what you can see is that if you use a reference control that is well matched on how the fluor is behaving in the multicolor sample, then the algorithm performs great and is going to give you accurate results. And finally, step number 6, check panel performance. No matter if the panel was already validated, take the time to check the quality of the staining, the pattern of expression of the different markers, to be able to identify underlying issues with staining quality. A missing antibody, lack of FC blocking, shorter incubation time, changes in antibody logs. So a good strategy is to visualize fluorescence versus side scatter for all the markers. Make sure you can detect a positive signal for each marker and that all signals are on scale. In addition to that, make sure that the results make biological sense. Use your controls, either biological control added to the data set or your internal controls. For example, if you are evaluating T cells, CD3, CD4 positives, and then define the memory subsets using CD45 array or CCR7, we get the 4 subpopulations. And if we further analyze those, let's say naive or effector memory for the expression of PD1 and CD27, we can use those populations as internal controls to define whether the staining works or not. So we know that naive cells, they don't have high expression of PD1 and a high CD27 expression versus effector memory where we see a lot of PD1 and the bimodal distribution for CD27. So use the controls as well to make sure the panel is performing well. So take home message, 6 steps to assess data quality. Your instrument performing well, your settings for acquisition optimal, your reference controls are high quality and that's reflected in your unmixing. You remove any unwanted events, especially technical issues or anomalies, and that you check the quality of your staining and the performance of your panel. So after you ensure you generate high quality data sets, the question is, how do I select an algorithm, pipeline, or a software for automated analysis? And there's been a search in the development of computational methods to overcome the limitations in manual gating, especially when the complexity for these data sets not only comes from the multiple markers in the panel that you measure simultaneously on each cell, but all the other variables in the study, multiple treatments, multiple patients, multiple sample types, thousands of cells, per sample. So as a consequence, we know computational techniques are necessary to clean the data, to visualize it, and also to interpret these high-dimensional data sets and make any correlations with clinical data. Some tools can also help with data cleaning, so you don't need to do it manually, and can be incorporated as part of the automated analysis pipeline. Some of them to detect and remove anomalies, especially irregularities in the flow rate or during signal acquisition. Some other metrics that you can use to evaluate staining quality and flag events or samples that have staining issues. And also, if you want to integrate multiple data sets that have batch effects, you could try to remove or normalize the unwanted variation, and there is algorithms for that with the caveats that they could have. So the goal with any data visualization tool is that you can extract the information about the different cell populations in a sample in an objective way, taking into account the expression of all the 33 markers in this case, or all the 33 dimensions in our data set. And there's multiple approaches for that. For example, you can do clustering, which is the process of grouping together cells that express similar markers and classify them into populations. Another example is dimensionality reduction, which is a low dimensional, 2 dimensions in this case, representation of the data that is reflecting the structure in high dimensional or in the 3-dimensional space. But remember that it doesn't identify populations, just creating 2 new parameters for this data set to describe the data and build this 2D map where the distances between points are going to reflect the similarities in high dimensional space. And there is multiple algorithms to do this visualization. For example, I'm going to describe here some of the algorithms and also what's the general workflow that you want to do to implement these tools. Pre-processing first, clean the data, transform, do normalization if needed and if it's working well. Then you want to identify your subsets, clustering multiple algorithms. You can then visualize those clusters or the marker expression using high dimensionality reduction, or do the opposite, start here and then go for clustering. And finally, use that information about cluster frequency, marker expression to do statistical analysis or predictive analysis using all the other information in your study or in your clinical trial. So remember that no matter what's the algorithm that you use, you need to make sure that that algorithm has the ability to find populations accurate, that has high computational efficiency, that remember that the optimal package that you use for each step should be selected, evaluated by the investigator based on the scientific question, and that significant tuning of the algorithm parameters might be required before you are confident in the final analysis. Really great that for some of the options we will see is that you can add metadata, sample information, clinical information, important to do or make correlations between the clusters and the clinical response, for example. An important last step is that after you get any findings using any of these algorithms, is that you can confirm the finding by using manual gating. Referring to our data set, it has 5 different donors, and we identify by manual gating that donor number 1, if you gave the T cell CD4 positive effector memory, this donor has high expression of CD56 and 57 as well as PD1. So important to verify your findings. Verify cluster stability, test multiple algorithms to compare if needed, and adjust all the hyperparameters in those algorithms to get high quality results also at the data analysis step. Good point is that you don't need to do it on your own. There's multiple software packages available to choose from. Some of them are open source within the R bioconductor programming environment or other platforms like MATLAB or Python. So you can freely access the scripts or pipelines needed to implement these algorithms, but it requires expertise in programming. Other options are driven by graphical user interfaces, like the ones we will discuss today. So now we are ready to listen to such a talented group of scientists from our data analysis partners. They are going to tell us more about their features, workflows, and tools offered by the software packages while exploring all of them their same data set from Cytek, which includes 5 multicolor samples, 5 unstained controls, and 33 reference controls. Thank you for your time and attention. It is wonderful to have you with us today. And please feel free to reach out if you have any questions. Thank you so much.
Okay, thank you, Deanna. There's one question that came up along the daily QC, daily tracking aspect of your presentation. We're wondering if you could explain a little bit when you're monitoring a laser's performance over time, what exactly are you looking for to know that things are working well, or when things are starting to decline? What should you be concerned about?
We can go back to that slide. And what you do when you run QC is you use beads that you know what's going to be their intensity of fluorescence across all the multiple detectors. So what the automated software is going to do is adjust the gains to match that target of fluorescence. A couple of things that you need to pay attention to is how much the instrument had to adjust the gains in order to reach the same target. So the gain change shouldn't be that high, and all the specifications will be at the end of your daily QC report. Another thing important is that these beads are fluorescent in all the channels, and then you can measure the spread of the positive peaks for that fluorescent signal. That spread that are quantified by the percentage of robust CV, that spread should be low based on the good performance of your optical and also your fluidics in the instrument. So those are 2 things that I would pay attention to, the CVs and the gain changes, as well as the good calculation for laser delay and area scaling factor. Once again, QC passing alone is a good indication that your instrument is performing well. In terms of the Levy-Jennings tracking, you want to make sure that it's not huge variations, increases in detector gains upon time. So you want to pay attention to gains across time and also the CVs, the populations should be really tight. These are beads, very homogeneous and very well characterized in terms of the emission profile and the intensity of the signal.
Okay, thank you, Deanna. We have one question about single stain controls, because in SpectraFlow, there's that feature where you can store them and reuse them over time. So assuming that you have a optimized panel and you've optimized your controls and you're thinking about storing them, what are the considerations for how long you can keep reusing them, or any risks you might encounter in reusing those controls that you should think about?
Very important consideration. Yes, the software has the capability to reuse controls that have been stored. But in order to do that, multiple things need to be confirmed. Your instrument needs to be working perfectly. You need to confirm that the QC pass, that the instrument is stable, that your signal across all the different detectors is stable, and also that the linearity between the gains and the intensity of fluorescence is maintained based on the APD functioning. That's one thing. And the other one is that the only way you can reuse controls is after you have done all the steps for panel validation. So you make sure that your controls give you perfect unmixing, no errors, and that your controls are matching, either if you decided to use beads or cells, that you confirm that if you can use beads, it's because the beads are going to give you good unmixing, no errors, once again, because there are differences between beads or the spectrum profile that beads can give you compared to cells. So make sure that before you store a reference control, your panel has been well validated, and that the controls that you're storing give you good unmixing. And in addition, whenever you reuse a control, because the panel is well validated and set up, and because your instrument is working well, make sure the unmixing is still accurate, that the populations are still behaving the way you expect without any errors. And I think it's critical in this case to have reference biological controls where you can judge things like that.
Okay, great. Thank you, Deanna. To keep things on track, I'm going to keep going and start introducing our next speaker. There's a few more questions that came up, but we'll answer those privately through the Q&A, and if there's time at the end, we'll take those live as well. So our next speaker is Chris from OMIQ. He's OMIQ CEO. Chris is originally a cell biologist and a cytometry expert and very passionate about flow cytometry. In 2013, he switched his career full-time from being a cell biologist to getting into software engineering and data science. He co-founded OMIQ in 2018 to focus on analytical innovations and accelerate the progress of human health. So let me toggle control over to Chris so he can demo what he has to share.
Great,
There you
thanks,
go,
Janelle.
Chris.
Can you hear me?
Yes, loud and clear.
Okay, great. And can you see me? Well, my screen at least.
You can see your screen and the lovely picture of you.
All right, so you can see me. Okay, great. So thanks a lot for that intro. And hi, everybody. I'm happy to be here today to talk about OMIQ. OMIQ is a new data science platform that we've developed for single-cell biology. Real quick, the executive overview of what OMIQ is that it's a cloud platform. And primarily, we've built it with cytometry in mind, but also recognizing that more and more these days, there's sort of an integration of different types of workflows and needs within quantitative cellular analytics. And so with that in mind, we also have the ability to upload other types of data and process them just as well. I think one of the unique things about OMIQ is that we like to say that we don't have very strong opinions about what data should be and what type of things you might want to do to data. And you'll see that idea playing out during the rest of the presentation and the demo. I want to give you a little bit of background about myself, more pictures, more color to my past than Janelle gave you. As she said, I was originally a cell biologist. And so when I approach problems in this field, it's really with that hat on. When I was a biologist, what I cared about day to day was probably what most of you care about, which is just getting to the next truth in the data, getting to the next experiment. And to me, analysis was just a thing that I had to do in order to get to that next step. And I always had the same problems with analysis. And that was that it took too much time, way too much manual labor, and way too much fragmentation was involved in that process. And when I talk about fragmentation, the one that was the most painful was moving in and out of different softwares. It made traceability and auditability of my workflow hard. It also made it such that when I invariably messed something up at one of the early stages and realized it at the later stages, I had to go back and redo my movements between 3 or 4 different softwares. So this is very painful. Fragmentation was also hard to get in front of other colleagues and collaborators, right? Just to have them look at the data. This is also during a time before we had high dimensional technologies, right? And the one that we're talking about today is the Aurora. Of course, we know about the Cytek and these sorts of things. And what I saw in my life as a scientist doing was just getting way harder day to day. And then, of course, we had this influx of new ways of looking at data, right? There was this introduction of high dimensional analytics. Myself at the time, and I think a lot of scientists even these days, don't understand this too well. It's often a novel and mysterious concept, but it's very important, right? It's not only useful, but necessary in order to help you get the most out of your data and not miss anything that's happening. And, when I was looking at this happening and the field changing, I was thinking this thing, which is analytics is going to be the bottleneck in the advancement of human health. And fast forward to today, we see this all the time, that analytics is the bottleneck in the advancement of human health. I think we see just across the board people spending potentially weeks or months with data sets that should be able to be analyzed in days. And so that problem is really what we focus on day to day at OMIQ. And our ultimate goal is to just solve data analysis. We want to make it into a solution instead of a process, right? And so that's a pretty hard problem to solve, but this is basically our steps toward doing that. The first thing to acknowledge is that we're interested in as much as possible as offering automated solutions, right? So instead of just having to go through and do all these different steps in different software, we want to have one software where you put in data and get answers out of it, right? Really what that means behind the scenes is taking advantage of all the new innovations in analytics to build smart pipelines to be able to process your data automatically as much as possible for you. But if you look at the field and where it is and what people need ultimately day to day and all the various needs in the field, what we also can't escape is that we need flexibility. When I say flexibility, I mean the classic forms of data analysis, right? It's not only about using algorithms and this sort of thing, it's also about getting in, interacting with your data manually, doing things like gating, interacting with those results, verifying them, and that sort of thing. And you can't really escape that. And so ultimately, recognizing at OMIQ is we're trying to blend these two needs and these two major patterns in data analysis together. So not only giving you automated analysis, but also that human element, right? To build this sort of bionic suit to help you use your expertise and pair that nicely with machine learning to get you the results that you need. Thinking again in terms of what we're doing with OMIQ and what you need in your analysis workflow, we recognize two patterns of analysis, right? So this is getting to like sort of the theoretical level behind what we're building. On the left, you have processive forms of analysis. So these include most of the innovations happening right now in programming languages. Deanna gave a great overview of these, and the need to be able to step through a sequence of analytical steps and track that. And that process is becoming more complex. And so this is sort of what we call the processive aspect of analytics. And then again, as I mentioned, we have to pair that with the interactive. So regardless of what you've done to your data, being able to at any point get in there, make plots dynamically, interact, visualize, really turn things over left and right and dig into your data. And so with OMIQ, what we've done is to put these two patterns into one place. We've built a modular architecture that allows us to plug in any sort of algorithms and this sort of thing and keep up with innovation. But then at the same time, we're not sacrificing that real-time data analytics and visualization that you're used to and that you need. Thinking about cytometry specifically, we can again sort of call out individual things that you're doing, right? So in terms of your classic analysis, we have compensation, scales, gating, making figures, doing batch reports of statistics and visualizations, heat maps, concatenation, stuff like that. And then we have these modern needs. So a lot of the stuff that Deanna talked about, right? So we have all your clustering and dimension reduction, often not as well used algorithms due to accessibility, such as trajectory analysis, ways of automatically cleaning your data, and then also getting to the big picture results, right? So it's not about dealing with one file at a time, making one plot at a time. We also have statistical methods that allow you to cut through all that information and pop out what's most interesting in terms of your clinical or experimental metadata, right? Across your whole data set and all your samples. And then of course, we're wrapping that all up into auditable workflows, right? Keeping track of everything that we've done in a workflow, every step, and it's all in one piece of software. And so that's really the theory behind what we're doing. And so at this point, what I'll do is stop talking about it from a theoretical perspective, and we'll just jump into OMIQ and see what it looks like, and we'll walk through a demo workflow. And so the files that we have here, as Deanna mentioned, were provided by Cytek. It's a 33 color sort of normal human immunophenotyping panel, and we have 5 donor samples, and we'll see what we can find out about those samples. When you are operating in OMIQ, keep in mind it's a cloud platform. All you need to do is find a browser or any web-enabled device, and you can log in and do your work. There's no installation of software, there's no installation of plugins or anything like that. When you're looking at a data set, which is really just sort of a folder for data, you see the files that you've uploaded. And what you can do is investigate those files one at a time if you want to look at their metadata. You can look at the stuff that was uploaded, that was present at the time of uploading. We store all data in a raw state and then build analysis on top of them. So we're never editing raw data for compliance and storage issues. You can also download individual files and delete individual files from a data set. When we're talking about setting up an analysis, the first thing that we want to do is curate file metadata. And this data set is not particularly interesting as far as I know with respect to metadata, but metadata are the type of information that you get from clinical studies or experimentation that you're doing on the files. We include those as columns of information. So you can see here I have my file names, and in this case I've actually put one column of metadata in, and I called it donor. And that donor column has values in it. And so you can accumulate as many columns of metadata as you want. You can upload them easily by spreadsheet. You can also import metadata automatically from files. So if you want to acknowledge certain key pieces of information from your FCS data, we provide that for you here to advance into your analysis as well. And then you can use embedded metadata automatically to help get good answers out of your data. After setting metadata, the next thing you want to think about is your channel names. And we call these in OMIQ feature names because we're more generic than just flow cytometry, where we normally say channel. In OMIQ, a nice thing about the platform is that you can change channel names, either the primary name, which is often giving you the instrument, channel or detector, or reagent, and then the secondary name, which is telling you usually what you were actually measuring on that channel. And so a common bane of the existence of any cytometry analyst is having some sort of error in channel names. In OMIQ, we don't really care about that. You can change it easily, and it doesn't matter what it is. I see that there's actually an error in this file. It looks like the live dead channel was labeled with the sample, maybe the sample numbers, as opposed to being an actual name. And so I'm actually going to not correct this. I'm going to leave it here because it's useful for demonstration purposes, how even when you do have conflicts, OMIQ will try to work around them as best as possible. And the last thing you might want to do before you actually dive into analysis is to think about who you want to collaborate with. So OMIQ has full collaboration abilities. You can add people in different roles, such as editor or reader, and you can send messages along with your request to collaborate and this sort of thing. Now we can get into the fun stuff, which is analysis. And so we'll see how analysis is structured in OMIQ by way of looking at this concept called a workflow. And so the way to start a workflow in OMIQ is to click the new workflow button, or the create workflow button. Once you create a workflow, we can immediately see this important concept with how OMIQ displays your analysis. And this concept is really kind of at the root of how analysis works, which is to acknowledge that all analysis is a sequence of steps. And so looking at our workflow, we can see that visually represented to us. And I've actually seen veteran cytometry people who aren't aware of what these steps actually are. And the fact that they're occurring in most software is behind the scenes without your express knowledge of it. And so one of the first steps, of course, is compensation. If your files have compensation in them, they'll be detected and applied automatically. Scaling, and then we can decide what we're going to do next simply by clicking on this button here. So if I click on this button, we can see that the main point of organization around how you do work in OMIQ is these lists of tasks. And we're adding to this list of tasks pretty continuously as new developments occur. And the benefit of our system is that we've done this in a modular way that allows us to keep up with the field. We have all sorts of options in terms of your basic processing needs, right? Scales, compensation, gating. We have different forms of dimensionality reduction, clustering, graph analysis, heat maps, trajectory, a lot of the stuff that I already mentioned. And of course, automated cleaning and pre-processing and statistical differential analysis. And so what we can do now is think about actually demonstrating a workflow. And to do that, we'll start with gating. The things that we can do with gating, there's not a lot of special stuff to discuss here. What I'm going to do actually to accelerate things is paste a template that I already have. And so we can see that I'm capturing just some basic cleaning of my data. We have singlets, live cells, and then a scattered cleanup gate. You can see that it's convenient to look at how you gated something by simply clicking on a population name. And there's also mechanisms that I won't describe right now where you can look at where all your files land, where your gate lands on every single file in the data set. And so that information is easy to display and show. We can of course do things like increase resolution. We have full support for Boolean gates and various operations like that. And given our limited time, for now, I'll just move on. We can then think about how we want to subsample our data. So once we've cleaned it up, we want to actually choose a population to use and then move on with our analysis. You can subsample in different ways, either choosing equally or in different numbers of events per sample. And from here, we can do something like start a different forms of unsupervised analysis. And so in our case, what I'll do is demonstrate a cool algorithm called t-SNE-CUDA. What t-SNE-CUDA is t-SNE on a GPU, essentially. So it's over 100 times faster than normal t-SNE. And this is the type of technical advantage that we have with OMIQ, that we can use algorithms like this. So we'll set this up and then we'll run it. And in the meantime, what we'll do is also set up some clustering. In our case, I'll try maybe a couple different algorithms. I'll run Park, which I think is a cool algorithm that was recently described. And then I'm also going to fork my workflow and try and see what kind of results I get by running FlowSOM, right? The classic. We can also run metaclustering at different levels of metaclusters simultaneously, which is a convenient workflow to do. And we can see that we're now processing a lot of stuff at the same time here. So we'll see if anything completes before I switch to my already completed workflow. And you can see that tsnikud is already done in 17 seconds, right? So that's displaying that speed. So what we'll now do in order to avoid waiting for this computation to finish is I'll switch out and look at a workflow that's already done, right? Or at least one that I've processed a little bit further. So this is identical to the one that I just did. And we can ultimately continue our workflow by looking at things like heat maps to see the results of the clustering that we did. We can understand the identities of different clusters and relate the clusters to each other, both in terms of which columns are most similar across all of the clusters and which clusters are most similar across all the columns. This is interactive and we can zoom into different levels, etc. There's plenty of different ways to structure different coloring and that sort of thing as well. We can also do things like look at graph analysis, right? So when we have something like a FlowSOM result, we can look at that. We can also import metaclusters automatically onto the graph. When we're interacting with this graph, it's quite fast to flip between channels, flip between samples, and look at it in different ways that are also supported by these box plots, which normally you need a lot more samples to make use of these. So maybe if I turn this off, you can see. We can also select nodes on the graph by way of choosing a gate. And if we use OMIQ's abilities for split screen workflows, we can actually pop open a new tab. And this is a core principle when using the platform. And in the new tab, when you open two tabs of OMIQ, they start communicating with each other. So if I do things like select clusters here, the visualization in the other side is going to update. And so now when I make changes on the left, you'll see the plots on the right update. Of course, there's a bunch of other visualizations we might want to make. So for example, in our completed analysis, what we can do is open up a figure here. Now what we want to do is look at some of our classic visualizations of our t-SNE results. So we'll open that up. And then we can do things such as configure these how we want and take advantage of permutation functionalities and previously copied lists of channels, then really rapidly drill into visualizing a lot of data at once. And another useful functionality when we're looking at data in this fashion is to do things like virtual concatenation. So you never want to look at one file at once. Usually you'll want to look at groups of files or compare files to each other. And so if we were to turn on a file name, and then we can see we can turn on virtual concatenation, and then we can look at multiple files at once and we can choose which ones to look at. And this pairs nicely with the concept of, I'll pare down this figure for a second, trying to compare files by way of looking between them. So what I'll do is switch this back into density mode, and then I'll use permutation to look at different files. And this is a great way to identify differences between files. And in fact, let's see. I need to do that again. And so now when we're looking at the t-SNE map between different files, what we can see is that we have this obvious presence of a large population that's in one and not the others. Right? And so we can also see other variational differences. We can switch the type of plot that we're looking at. Maybe make these a little bit larger. We can switch into contour plot mode. We can also do things like I'll create overlay plots, and I'll turn this into a overlay scatter plot and overlay the different files with each other. And we can see if any interesting differences pop out. We can also set all of the layers to a certain color and then only select one at a time to see where the interesting differences are for that one file. And colors are kind of hard to see in this case. We can also, as a sort of final switch, do something like contour overlays. And this might get a little bit messy. Let's see what happens. Yeah, that might be a little bit hard to deal with. But the idea always that we're looking for is interesting differences between samples. Given this example data set, we're a little bit limited. There's only 5 samples to look at. We can, of course, dig in all these different ways using either automated tools or manual analysis. When you're thinking about using OMIQ, you usually want to get the most advantage out of it, a larger data set that's sort of clinically interested. So I'll show you some quick shots about what you can do with that. We have things like statistical analysis with volcano plots, looking at differential statistics and finding the clusters that are most significantly different between large groups of samples. So here we have 7 GVHD patients, and we're finding which clusters are most differentially abundant between the two. We can do things like look at box plots, and we look at box plots of all of our samples and automatically segmented by metadata and that sort of thing. And we can interact with these how we want. So we can turn on multiple filters, we can zoom in and interact. And of course, we can render these as things like violin plots and that sort of thing. So I think at this point, I've thrown quite a lot at you. The core principles that I'm mostly trying to show are that OMIQ blends the possibilities of automated analysis with those of manual analysis, and you can interact with those two very fluidly, right? And at this point, I think all we would have left to show is more and more different features and more different use cases of the tools that we have. And so I'll leave that maybe for you to try on your own using our free trial and our tutorials. In terms of last things to talk about, in terms of things that we do, one of those is custom tasks. So OMIQ is designed to be able to take in scripts in any programming language. These could be your own that you want to share publicly if you're a researcher. They could also be, if you're an enterprise organization, things that you normally have to run with your bioinformatics department or something like that. We can add those into the platform and run them natively. This also includes auto gating, right? So we're able to set up these algorithms, but also if you already have these, we love to just pop them into the platform and you can run them natively there. And so contact us if you're interested in that sort of customization. And what the only thing I want to do is thank you for your time. And at this point, I'll take any questions and what time that remains.
Okay, great. Thank you, Chris. So I shot over a bunch in the Q&A window for you. One of them is, does the collaborator need to have an OMIQ account?
Currently, yes. So, there's kind of no way around needing to have an account, but we do have functionalities for reporting. So that's one thing I didn't show, is that you can very easily sort of make any figure and then export it as a PDF. We also have this interesting batch reporting tool. So if you've done a very large analysis across a lot of samples, you can export, for example, like a 50-page PDF with all of your analysis done automatically. We do offer collaborator accounts and flexible licensing schemes for this sort of thing. So you'll just have to get in touch with us for your situation.
Okay, thank you. Could you elaborate on what the AI function does?
I think this might be Flow AI that we're talking about. So Flow AI is a data cleaning tool. So we can look at that here. So normally you'd run Flow AI earlier in your workflow. And what it does is to find the regions of your data that are, well, the cleanest, right? So it'll remove anomalies either in flow rate or outlier signals and that sort of thing. We have a whole white paper on Flow AI, so you can get in touch with us about that.
Okay, great. Another one, there's a few questions around file size and size of experiments. Specifically, how does OMIQ handle the simultaneous analysis of big data files on the order of hundreds of megabytes? And these experiments are meant to identify rare cell subsets. Do you have to do a lot of downsampling? Does it take a long time to compute? If you could speak a little to that.
Got it. So there's sort of two ways to think about that. The first is for any sort of real-time processing, so gating, generating plots, that sort of thing. And the second is algorithmic analysis. So in terms of real-time processing, the answer is that we've had to do sort of a lot of careful engineering to make sure that OMIQ is high performance no matter what. And so we have some users that are analyzing, for example, hundreds of millions of cells in a data set. That's, I think, the largest that we've seen at this point across, something like 100 plus files. We also have different algorithms that can accommodate different levels of data. So this is something that's harder to solve. We've worked on doing stuff like that through our work on Optisne, for example. But there's no getting around the fact for now that you will have to choose the algorithm according to how much data you have. So if you have 100 million observations, you're going to be much more limited. You might have to downsample. You might have to choose your algorithm appropriately. And so, our documentation or our support crew can help you make those decisions as well.
Okay. For the Tisne CUDAs that you showed, what makes them so much faster?
The reason that they're so much faster is because the instruction set for the algorithm has been mapped to process on GPU, right? So GPUs are graphics processing units. It's a special form of computer hardware that's used for rendering graphics, but you can also take advantage of them for other types of machine learning that does a lot of computations at once. So there's a lot of literature on the internet about this. So for further discussion, I'd put it there. There's also a paper for Tisne CUDA. It's an algorithm that came out of Berkeley, I think a year or so ago. Definitely a good read, so check that out.
Kind of related to the Tisne CUDA is, can you comment on the reproducibility between runs for that algorithm?
So this is either an advanced question or a basic question. So Tisne CUDA is interesting because this algorithm actually does not have a reproducible seed. We've looked into this. We're thinking about trying some stuff out to reproduce, to make that different. But that's actually unique to Tisne CUDA on this platform for the most part. You'll notice that anytime you're configuring algorithms, we offer you, well, this is a bad example because it's already seeded, I think. Different algorithms that you'll run will have the option to include a seed. And so the seed is what controls reproducibility. And so almost all algorithms on OMIQ, if you run them twice in a row with the same seed, you're going to get the same exact results, right? Certain algorithms, we've even fixed bugs, such as the original implementation of SPADE has what appears to us to be a bug in it that makes it impossible to reproduce. And so we actually went backwards and fixed that algorithm and offered it a seed. So, reproducibility is obviously very important to us. And so random seed, whenever you see that, it's reproducible.
If I want to get OMIQ on my system, what are the system requirements that I need to consider?
There's basically none. Because OMIQ is a web platform, all you really need is a basic computer. You can even hack it with a tablet or a phone. All you need is a web-enabled device. And beyond that, it's all sort of nice to have, I guess. It's not going to make much difference.
Okay. When you run the Tisne, does it create a new calculation of Tisne with all files included, or just a separation of the already concatenated file with the Tisne results?
So I don't know if I understand this question perfectly, but it's a good generic point of discussion, which is in OMIQ, we take care for you of concatenating data for the purpose of running an algorithm. This is always how you have to run an algorithm for most algorithms. You have to concatenate the data, and then behind the scenes, we actually split them back out again for you to do. So you don't have to concatenate data as like a manual step to run algorithms. And when you do virtual concatenation, it's only for plotting and other sort of dynamic things. So what we do is we virtually concatenate in real time on our server and then display the data to you in the concatenated form. And so usually that's the convenient way to do it. You don't want to use, we have permanent concatenation as well. Normally you don't want to use that except in cases with like where your sample was so large that you had to acquire it over multiple tubes or something like that. And that properly it should be concatenated. For the rest of the time, you just want to use virtual concatenation.
Okay, I'm going to give you one more, Chris, and then we'll move on to our next speaker. Since this is a cloud application, what security protocols are in place to protect the data, and what does a local IT group need to consider to allow access to the system?
Yeah, so we use what I should say is a sort of enterprise standard security. This involves organizational security. So we have a lot of, at our company, a lot of protocols and documentation of our security standards. And then of course, on the technical level, we use end-to-end encryption in flight and at rest. We use all sorts of different virtualization technologies to make sure data are separated, when it comes down to that. If you're thinking about an organization, usually for companies, you'll have an IT group and they have standard questionnaires. They'll give us this questionnaire, we'll fill it out, and then we'll be good to go. So there's standard processes for that at most companies. Most universities, it's very different. Some or any academic group, some don't seem to care all that much, or it's just sort of make sure the basics are in place. Others have their own IT groups that do audits of our security standards. So I hope that answers the question.
Okay, great. Thank you very much, Chris, for your presentation. Anyone's questions we didn't get to yet, don't worry. We'll have more time at the end, and also we'll keep writing back and forth with you privately in the question and answer box, no problem. So our next presenter is Sean from Novo Software. He's a senior product manager there. Sean has been working to advance data analysis solutions in FCS Express at Novo for the last 10 years. Prior to working at Novo, he utilized flow cytometry in the study of interactions between HIV-1 viruses at NYU and research methods for automated microscopic analysis of tissue sections at the single cell level at Memorial Sloan Kettering Cancer Center. Let me toggle controls over to Sean, and Sean can take it away.
Yeah, hi Janelle and everybody. Thanks for having me on the call today. Let's see here. I might see if I can turn on my webcam too. You can see me, and let me pull up my screen here. And are you seeing the screen come through here, everybody? Does that look okay?
Yep, looks good.
All right, great. So let me get some stuff out of my way here, and we'll pull this up. All right, great. Yeah, so thanks again for putting this together, the team at Cytek and Janelle in particular. It's really great to hear about a lot of these different types of tools that are available for folks to perform high dimensional data reduction. So again, my name's Sean Burke. I'm the Senior Product Manager at Novo Software, and what we're gonna be covering right now in the next half an hour or so is solutions in FCS Express for high dimensional spectral flow cytometry data. And where I'd like to kind of start with this is, why use FCS Express? So I know a lot of folks out there are already using the software, but if you're not, why would you even, want to bother? And especially for working with high dimensional data sets. So I don't know if there's anybody out there like me. I used to actually own this Nokia phone. It was actually the first cell phone that I had, but it really did one thing. It made phone calls. You could also send a $1 text message if you wanted to. Maybe it did three things. You could play a game of Snake, right? But all of these technologies kind of have that same type of thing where you would perform one sort of, you take a picture with a camera, you take a video with a video camera, a GPS, and right now everything is kind of condensed into a smartphone, and that makes life very, very convenient. And that's also kind of where we're at with the need for convergence with flow cytometry data analysis. It's probably something you heard a little bit from Chris in the previous talk as well. There's all these kind of tools that folks generally need to perform analysis. And what we do in FCS Express is we bring that all together. So imagine having the power of Excel directly related to your gates, the power of Prism directly related to your samples, things like PowerPoint, cell cycle modeling, and really all of that is wrapped into FCS Express. So literally as you move a gate, all that kind of downstream analysis gets performed in the software. And that's, again, the big difference between our software and other software packages that are on the market. When you're working with other software tools, you may be getting basic plots, you're getting tables of statistics, but more often than not, you're copying and pasting and moving those things out to Prism and to Excel. I've seen people do really complicated things in Photoshop just to put a little white text box over an axis label, and also having to use additional tools like R. If you don't have some sort of programming background, it can be difficult. All of that before you get to a result. And what you're going to see today and what we're going to talk about is how we can get those results in FCS Express. So working with the raw spectral data, creating bar charts and pie charts, working with t-SNE plots, all of that type of stuff is wrapped directly into the software so that you end up with a final result before you get anywhere else. And before I move on, I heard a little bit of background noise. I wonder if there's somebody that might be able to put themselves on mute there. But to kind of come back around to FCS Express for high dimensional data reduction tools, one of the problems is too much data, and it's not really a problem. We want more data. You have this awesome spectral instrument from Cytek that can collect all of these parameters, and in the past, this is kind of mind-blowing, right? You look at N by N plots of everything, and it seems a little crazy. So there's a few solutions. Really, you can analyze the data by looking at 2 dimensions at a time, kind of using your intuition as a scientist to create gates and interpret what that means. There's other tools that do really complex math in some sort of n-dimensional space to get out an answer. And then there's another way of trying to visualize data on a single plot and trying to figure out and interpret that plot. And what we're going to cover today is essentially these 2 solutions. And I think it's something you heard from Diana in the beginning, you heard from Chris a little bit as well, that you can use these in conjunction. You don't have to use just one or the other. You can use kind of classical panel design, you can use high dimensional data reduction tools, and both of those being pulled together are going to give you a better understanding of what's going on with your data. So what we're going to cover today in FCS Express is essentially what I call classic. It seems like it's old-fashioned at this point with all these algorithms that are out there to perform analysis, but it's still very powerful for working with large data sets like this. So we're going to look at some of the integrated and real-time statistics, the cohesive graphing and charting and spreadsheets in the software, and how we can utilize batch processing for really great efficiency for reporting information from many data files and parameters. And then we'll take that a little bit beyond too. We'll go into the high dimensional data analysis tools, which are incorporated directly into FCS Express, and a really easy to use interface. These allow for full exploration of the data with some of the fastest native calculation times on the market too. And the really cool thing about these in FCS Express, you can really easily move between classic panels and high dimensional tools to explore that data. Now, the other component that's very unique to FCS Express is that you can access that raw spectral data at any time, right? You've gone out of your way to get this awesome instrument from the Cytek folks. It has all of the raw spectral data and spectrum signatures associated with it. And in FCS Express, you don't have to throw that away and just work with the unmixed data set. You can actually access the raw spectral data, and you can even do spectral and mixing directly in the software itself. So we'll kind of stop talking about that and we'll jump over and take a look at some data and give you our take on this 33 color panel. And I'm going to open up FCS Express here so we can start having a look at what's going on. And you can see that when I open up FCS Express, you're probably like, hey, Sean, you're still in that PowerPoint presentation. Well, shamelessly by design, we have essentially used the Microsoft Office look and feel for FCS Express. And that means if you want to insert text boxes and shapes and pictures, you can do all that. But this is also how we access all the plots for our data. So if you've ever used PowerPoint, if you've ever used Excel, you're already an expert at using FCS Express. And in this, what we call a layout that we put together, the first page here is an experiment overview. And this is just essentially a big image, right? And it's showing us what we're going to be analyzing today. But it is a very powerful tool, right? Anybody who opens up this panel, who's going to look at this data in this kind of classical panel format, they're going to know what you're looking for before you even move on to anything else. Now, another kind of nice tool in FCS Express is the concept of using a page for reporting. Now, all of these are appearing blank, but if I choose a data file, here it's the 5036 donor, what FCS Express is going to do, it's going to load it up into this analysis template, and it's going to report all of the reportables for this particular panel. We got some nice little images in here showing what these are as well as pull in all of the keyword data. And again, the way that FCS Express works is that as we move between data files, all of the information in this kind of report page is going to update. Once that new data file gets loaded into the software, you can see that these things update. We can switch back to the previous data file and we get the information associated with that. Now, where are these statistics coming from, right? They're coming from a lot of different pages. And when you work in FCS Express, just like when you work in PowerPoint or Excel, you can have different tabs, different slides for analyzing things. And what you can see here is that, on this page, we've set up some of our kind of basic scatter plots. We have a page devoted to our monocytes, and we have really pages devoted to all of the analysis we're performing. But what you're going to see is that on some pages, the important information, things like the breakdown of the CD14 and CD16 populations of our monocytes, they're in a pie chart. And that means that as we move a gate in the software, as we move a quadrant, as we make some sort of change, the information highlighted in that graph updates, right? It means that we didn't have to copy and paste out to Excel to get that information into some sort of graph. And that's kind of a recurring theme in the software that you're going to see is these live updating charts, which are essentially final reports, right? And when we look at our TCR gamma delta cells here, this is just an example to give you an idea that you don't have to work with just density plots. You can use contour plots with black and white stripes. You can use color backgating. There's really just about every sort of visualization that you used to be using, or that you've seen published on the market or published in publications. It's all here. So as we kind of go through a few of these pages, I'll just highlight a few things. Again, we have our B cells that are linked up here. We're looking at our IgD to CD27 ratios. And again, it's not just the gates. As we move between another data file, that data file is going to come in. We're going to see this information update in the software, and we're going to get some real-time results about what's going on with these. Now, the charts don't just have to be pie charts. Right here, we're looking at our dendritic cells. We have our PDCs and our MDCs. And again, as we kind of move these gates around, we get that live updating information in these charts, which again is an end result. This is something we'd have to do in other software, bringing it out to tools like Prism and GraphPad to actually arrive at that. So again, it's a big panel, right? There's a lot of pages here, but you can easily break them down into manageable chunks to do some sort of, kind of classical analysis. I know there's a lot of focus on using high dimensional tools, but these tools for kind of classical analysis are still very powerful. And there's something that I think should be used in conjunction with the high dimensional data reduction tools, which we are going to jump into here in just a second. But in the end, we really do focus on that end result in FCS Express. And here's a nice little graphical report of just a few of the subsets, right? Here's our B cells, here's our monocytes, our dendritic cells, here's our CD8 effector versus CD4 effectors for this particular sample. And again, if we come back to something like maybe a two-page view, and on one page, we have our graphs, and over here, we've got some of our gating. Well, if we change our live dead gate on the first level of this hierarchy, what we're going to see is, just like, half a second here, everything, including all of our graphs, are going to update. And if we need to say compare, right, let's look at donor 4559 versus 4604, we can change data on that particular set in the analysis. And we're going to see that all of our graphs downstream are updating, and we can really kind of focus on, again, that end result. But when you're running data analysis, or when you're running samples through your instrument, you have a lot of parameters, but oftentimes you're going to have a lot of samples, right? In this case, we just have 5. But if you want to kind of crunch through that data very quickly and easily, it's very simple in FCS Express. We use something that's called batch actions or batch exporting. So if we need to get certain aspects out to PowerPoint, certain aspects of this analysis out to Excel, out to a PDF report, essentially what we can do is add these actions to this list. And just so you see here, I know this is a lot of kind of stuff going out to Excel. The way that we populated that is we just grabbed all of these text boxes and dragged them in there, and it populates our Excel report. So what would happen is if you have 500, 50,000, doesn't matter, you click run on this batch process button, and FCS Express is going to crunch through the data that you ask it to crunch through. So it's doing two of two things. It's exporting to PowerPoint, it's exporting to Excel, and it's doing that across 5 different data files. Now, as an example of what this looks like, I've already done a little bit of the export to PDF here. So you can see that these come out as high resolution graphics. These can be really whatever resolution you want. You have everything that you need. In this case, we exported a full report for every single sample. So for all 5 samples here, we have separate PDF reports that are being exported that, again, give us the full report, all of the pages, 13 pages of the analysis. But in reality, all 13 pages of this are probably not most useful for kind of collaborating with colleagues and kind of distilling that information. And what we've done, and what you're going to see here in just a second when this batch processing gets done, we are running this in real time, is that PowerPoint is going to launch up and Excel is going to launch up. And then we have all of our reportables, right? We know that FCS Express is not the be all, end all. And if we want to get some information out to Excel, like all the reportables for this big panel, we can do so. And additionally, we have all of this out to PowerPoint. And when I say all of this, in this case, we just wanted the report and the graphic report, right? So we have the report and we have the graphic report, 10 slides, 2 slides for each of the data files. And the really cool thing about FCS Express is these PowerPoint exports are all individual, what's called vector graphics. So if I wanted to group them and ungroup them and bring them to the front and move them to the back, if all I needed was this B-cell distribution for my poster at Cyto, you have all of that here, right? And that's a result as well, being able to take your data, share it with the community at a conference or poster presentation or publication. FCS Express really helps facilitate getting these results quickly and easily, either through using classical panels, through doing these kind of batch exports. It's all there and pretty accessible. And again, if you know how to use Office, if you know how to use PowerPoint, you're already halfway an expert. Now, one of the focuses for today's talks is talking about high dimensional data reduction, right? So, classical panels are just kind of one way to do that. We're reducing the data that we have into essentially a report page. But if you want to explore this more, you can see here that we've set up an analysis. It's a little bit smaller. We just chose a little bit less pages, so you're less focused on the pagination here, and I want to get into how t-SNE works in FCS Express, right? So you can see here that we have some t-SNE plots inserted, right? We're looking at t-SNE Y versus t-SNE X. We're looking at a contour plot up here. We're looking at a density plot. We're looking at a color dot plot. And how we perform t-SNE is that in our transformations tools here, we have the ability to add a number of different types of clustering and transformation tools. You're actually going to see a lot more in just about another month or 2 here. We're going to have a big release of FCS Express 7 that has more tools. And what we've done is essentially said downsample to one of the gates. We've merged a bunch of the different data files. We've selected which parameters to run in this, chose a sample size. We chose to use OPSNE. We've chose to upsample the events. So again, we could talk about the variables in this for probably a whole other hour, but really the idea is when you perform that t-SNE, you get it applied to a plot, you're working with standard types of flow cytometry plots to help explore that data. Now, one of the kind of common visualizations for t-SNE plots is just using color backgating, right? If we want to say move between this plot and identify where just very simply the lymphocytes and monocytes are, well, down here, the lymphocytes are in purple. And what will happen is as we move that gate, you're going to have live updating on that t-SNE chart of where the monocytes, or what we're defining as the monocytes, or what we're defining as the lymphocytes are. So that live updating, once you run your t-SNE calculation, that same thing applies throughout whatever you do. Now, we do realize that when you run t-SNE, you're going to be looking at, all of the parameters that are here, and you're generally going to want to look at t-SNE plots for all of this. So what we do is we have this kind of handy-dandy multi-plot tool here. If you say color by all parameters, what you'll end up with is a distribution of plots on the page showing you t-SNE X and Y across all the different parameters in the data set. So you can see here what we have is we have t-SNE X and Y, and we're displaying CD45, we're displaying HLA-DR, CD14, and we're doing this for, again, all of the different parameters in this data set. We've also inserted what we call a heat map, and this is one way that if we need to deconvolve the samples, right, we had 5 samples merged together, how we can do that, and I'm going to come back and show you that in just a moment. But the idea, again, is being able to move between standard flow cytometry, kind of a classical panel analysis, and using these exploratory tools. So what you can see here is that we've set up a plot here, and we're looking at TCR gamma delta, and we've actually set up a TCR gamma delta gate on one of our t-SNE plots, which is over here. And what we can do is just very easily confirm, right? I'm going to move that away from this TCR gamma positive population. And it's very easy to discern using FCS Express that this indeed is a TCR gamma delta positive, right? The same thing goes here. If we look at where we had a plot that we were using to identify our CD8 cytotoxic T cells. Well, again, we can go back over here to our CD8 t-SNE plot, and that plot on the right is going to show us what the phenotypes of these populations look like, right? So again, it's very important to kind of have the ability to move between these types of plots in real time. And just to kind of drive that home, here's another example of a plot type that we have. It's called parameter overlays. So in this case, we're looking at CD3, CD4, CD25, CD127, and really these are the parameters that we need to identify Tregs, right? And if we maximize this plot, this is another kind of nice little trick you can use in FCS Express. What you'll see is that these are linked up to this particular gate. So if I move this gate around, I can get an idea of like what these kind of these CD25 populations look like in here. But the hard kind of thing to figure out is we need to find our CD127 lows to identify Tregs. So I'm not quite sure where that is right here. I know that there's some heat. So one thing we can do is we can always change plot types. So if we grab that plot and we say, let's make this into a contour plot, well, all of a sudden, instead of viewing heat, we're looking at kind of counts, and we can see indeed, all right, there's 2 populations here. So it's probably one of these. It looks like our CD127 negatives. This probably looks like our CD127 lows, and it really helps us identify and facilitate getting to the bottom of these exploratory plots and results. But again, when it comes down to things in the end, we are going to want to compare between different data files, right? That's one of the whole points. We ran this t-SNE on everything, but we want to be able to deconvolve and say, all right, for each of these individual donors, of these 5 donors, what do the t-SNE charts look like? And this is kind of a little example of that, right? So what we've done here is we've used this heat map and we've said, create some gates on this. So this is the sample 1 gate, and it actually consists of 2 samples. But you can see that as I move this gate, as I click on different wells, this plot on the left here is updating because it's showing us the events that are derived from this sample. And we can group samples, we can pick different samples, right? There's a lot of information that you can derive from this. But you're also going to see one of these little bar charts over here updating, right? So this is actually showing us sample 1, 2, 3, 56B and T helper cells. And we can see, all right, well, how do these distributions change as I select different samples, as I select and include different parts of the group? And again, I know I've talked a lot about these graphs and these spreadsheets, and you're probably wondering, oh, why hasn't he shown that? It's probably really difficult to do. Well, here's a spreadsheet, right? And you can see it looks just like Microsoft Excel. In fact, it has all the kind of functionality that Excel has. And what we do to populate that spreadsheet is we say, all right, well, here's our B cells gate. This is our 20 positives, 19 positives as determined from our t-SNE plot. We can drag that gate in there and tell FCS Express, give us the percentage of gated cells, and that fills out that spreadsheet, right? And if you want to create a graph, just like in Excel, you'd probably grab a range of data. Well, let's insert a box and whisker plot for that particular spreadsheet, and then all of a sudden we have a box and whisker plot comparing group 1 versus group 2. And again, the nice thing is, as I change that gate on the t-SNE plot or anywhere in the analysis, those bar plots, those box and whisker plots, whatever it is you might be analyzing is going to update for you in real time. So again, you get that really powerful marrying up of the kind of tools that you would classically use downstream directly in FCS Express, which is very, very nice. Now, again, we maybe have lost sight of what this data actually is, all right? So we've been all looking at this like a standard flow cytometry experiment, but this is much more than that. This is a Cytek Aurora data set. It has full spectral capabilities, right? It has spectral signatures for every single cell that's in this data set. And FCS Express has a unique capability to actually access that raw spectral data. And what that means is that if we grab one of our data sets and say insert a spectrum plot for this, we can draw a spectrum plot just like an object in Microsoft PowerPoint. And FCS Express is going to allow you to plot the full spectral information. Lines or density plots for every single event in this data set are going to be plotted. And if we want to look at the full spectrum, this kind of just defaulted to the UV laser, but you can see that this is the full spectrum of the data. And right now it's ungated, right? This is just the raw spectrum. But if I wanted to say, all right, well, let's look at the spectrum from the monocytes. Well, I can drag and drop that gate. I can apply it to the spectrum plot. And that's now showing what the monocytes spectrum looks like. We can apply this here. We can see what the lymphocytes spectrum looks like. If we want to kind of drill down and get into, say, these CD4 naive cells, see what that spectrum looks like, right? We can actually go in and do that for any of these populations. You can create overlays. You can create multiples of these plots. But I think one of the really unique things, and I'd like to put this as a challenge to folks that are out there, try running a transformation, because one of the cool tools that we can use in FCS Express is we can say, all right, well, let's run a t-SNE, and I'm gonna set up a very small t-SNE here just so we can run it super quickly. We'll just grab a few parameters. We'll choose a small sample size, and again, we'll do an upsampling, and we'll use OptSNE, and I'll apply that t-SNE to the spectrum plot. So what that's going to do, it's going to run the t-SNE on the full data set. And when this says transformed in the title or t-SNE map, it's actually been transformed. And one of the kind of unique things that we can do in FCS Express is if I want to get that t-SNE applied and I want to look at, let's say, t-SNE Y on this, and we'll see what this clustering did. Again, I'm just kind of doing this off the cuff. We can see that this is a very basic transformation. But the really powerful tool that I'd like to see some people playing with is being able to set a gate on a t-SNE plot and then investigate on the spectrum plot, what does the spectrum of that clustered population look like, right? And that's something that we can achieve in FCS Express. And I think that's something very unique of the tools that we have here. Is again, you can move between the t-SNE plots, you can move between the spectrum plots, and it will be really nice to see what comes of these tools in the next couple months or years, once folks start being able to use these high dimensional tools, link them up to the spectral data, look what the spectral fingerprints look like for clustered populations. It will be really interesting what comes out of that. So that being said, I know we covered a lot. Half an hour isn't a lot to talk about. A lot of time to talk about a lot of these advanced tools, right? But again, it was a very kind of unique data set to work with. And some of the tools that you'll be using in FCS Express are classical panels, right? Just because it seems old-fashioned doesn't mean it doesn't get the job done, and it doesn't get the job done very well, right? We can use these tools to batch process through many hundreds or even thousands of samples. The high-dimensional data reduction tools are there for you, tools like t-SNE and SPADE. And again, keep an eye out on the Novo Software website. We're going to have a bunch of new high-dimensional data reduction algorithms coming in just the next few months. And importantly, having the ability to access that raw spectral data, right? You've gone through a lot of steps to set up for collecting this spectral data. Well, you can always go back and access that with FCS Express. If you wanted to do spectral and mixing in FCS Express, you could accomplish that now as well. But that's a whole other conversation for a different day. So some additional resources, FCS Express does come with a 1-year license with every Cytek Aurora system. So if you're already a customer, you should already have this. If you're thinking about buying the Aurora, we really recommend you do. You're going to get FCS Express with it, which is a nice little add-on. If you're not using the software yet, you can get a free demo from our website. And really everything that I've shown you today, the demo is fully functional, right? You can work with spectral plots, you can do Tisney and Spade, whatever it is that you want in that demo. Importantly, also on the Novo Software website, we've had a very close relationship and partnership with Cytek. So we have a lot of joint resources up there. The web link is here, but very quickly, if you just search Cytek on the Novo Software website, you're going to be brought to a bunch of application examples, short kind of how-to videos, 3 to 4 minutes, written tutorials. This webinar, the recording will be up there at some point as well. So there's a lot of information for you to go and gather. So with that, I hope everybody learned a little bit more about FCS Express and some of the unique capabilities and how we work. Hopefully your minds are not going to be blown by high dimensional data analysis anymore. And with that, I'd like to say thank you for listening to us talk for a little while here. Thanks for coming out for the first half of the webinars. And I'm happy to take some of the questions that have come through.
Okay, great. Thank you, Sean. Before we go into the break, let's go ahead and do a couple of those. One is, can you use and link your data to statistical analyses on FCS Express? For example, if I perform a t-test comparing the data on your bar graphs.
Yeah, absolutely. So just as kind of I mentioned, FCS Express looks and feels and works like Excel in a lot of ways. The one thing I didn't show in here today is all of the kind of functionality. There's t-tests, there's all sorts of comparative tests that you can run, and they work just like Microsoft Excel, right? So if you use the formula in Excel, if you wanted to do a NOVA, a t-test, pairwise tests, all that functionality is there. As long as you can get the data to the spreadsheet, you can essentially run the tests and you can graph that and report p-values, ANOVA, things like that pretty easily.
Great. Next question. Can the percentage of a particular population be drawn on various upstream hierarchical ancestors? For example, can we get percent CD4 effector memory of percent CD4 T cells, of CD4 positive T cells, and CD45 positive cells?
Yeah, absolutely. So, I mean, by default, FCS Express supports percent gated, percent of parent, percent of grandparent, percent of great grandparent. If things are starting to fall out sort of those norms, you can calculate customly any sort of kind of Boolean gating combination that you want. And don't forget, you have the power of spreadsheets. So if you drag in the percentage of the number of events for one population and the number of events for another, you can just use it like you would in Excel to create a new kind of custom ratio to calculate a percentage of any amount of anything else. But again, a lot of that's available just by default.
Okay, another question is, what tools in FCS Express would you suggest for users that are looking to evaluate their panel performance and evaluate their controls?
I mean, there's a lot of just built-in type of things. I mean, just visualizing plots, using time parameters to look at quality control. But in the near future, there's going to be direct integration or direct tools for things like Flow Clean and Flow AI. Again, that's coming in this release that we're going to have in the summer. In the past, folks have used the R integration in FCS Express to run kind of data cleanup tools. But again, a lot of the classical type of cleanup that you've been doing just simply with Gates is there. And if you wanted to unmix your data again, I mean, if you found that there's some unmixing errors and you didn't want to go back to spectral flow for some reason, you could actually do some additional unmixing in FCS Express, which might help you kind of figure out what's going on with some of the data.
Okay, great. Let me end on this one for now. We'll take the rest later. Is it possible to overlay the spectra from different subsets?
Yes, absolutely. And it's one thing I just didn't show just for the interest of time today, but that density plot that I put up for the spectrum, you can turn that into a line plot to just show the average. You can show line plots for the average of different populations. For as many populations, you can show averages for different samples that are in there. So really any sort of overlay that you want to accomplish, whether it be gates, whether it be files, you can accomplish any of that overlays with the spectral plots directly in FCS Express.
Awesome. Thank you so much, Sean. With that, let's go into our break. We'll come back about 10:45 Pacific time, 1:45 for those on the East Coast, and we'll resume with our next speaker that I'll introduce at that time. Thank you all. Okay, we are back. Hopefully everyone had a nice break. I'd like to introduce our next presenter. Her name is Beth. She earned her Ph.D. in molecular immunology and biochemistry from the University of Maine and continued research into inflammation and lymphocyte development as a postdoc at both the University of Maine and the Maine Medical Research Institute. Beth has over 20 years of experience in flow cytometry and research methods and analysis. She joined Verity Software House as an application specialist over 8 years ago and has since enjoyed providing customers with solutions for their high-dimensional data analyses. So with that, let me toggle the controls over to you, Beth.
Thank you, Janelle.
Here they come.
All right. Okay, so you should be able to see my title page and hear me.
Yep.
Okay, thank you.
Everything looks great.
Great, thank you, Janelle. Thanks everybody for joining us. So we understand that there are so many different analysis programs and algorithms to choose from. And there's also a great deal of confusion about the best choice for analyzing high-dimensional data. So we would like to offer you a solution that can not only analyze your data, but give you a whole different way of looking at it. GemStone has a revised user interface and added features that provides you with a comprehensive package that will help you get the most out of your data. GemStone features a multifaceted approach to data analysis. So step-by-step wizards will help guide you through building a template model using your panels and your experimental system. Templates can then be used for any number of data files using the automated batch system. And there is no limit to the number of measurements. So more measurements are actually better as it provides the program with more information to work with. Data cleanup can be automated and based on objective equations. And once a model is built, it takes only just minutes to analyze a data file, and that includes the Sensei graph calculation. Multiple users of that template will get the same results, which makes it a very effective tool to share with your collaborators. The driving force behind GemStone is called probability state modeling, which is a published, patented, well-tested algorithm that eliminates the need for subjective gating. Sensei, which is GemStone's high-resolution dimensionality reduction algorithm, is calculated independently of the model in an unsupervised manner. Sensei automatically labels the cell type populations and subsets, as well as providing a consistent orientation from sample file to sample file. All cell types are clearly defined with subsets and subpopulations of interest. Unique overlay plots display correlated measurements and provide a clear measure of comparison from sample to sample. Reports are constructed automatically and are fully customizable and editable. All graphics are also saved automatically during the analysis process in your choice of format. A full database is calculated and exported for further analysis. And you can also set up smaller tables on the report and populate them with custom equations, alerts, QC metrics, etc. Tricon graphs, which are not shown here, are a powerful tool for enumerating rare populations, unknowns, activation markers, and cytokines, whether singly or in any combination. There are built-in quality control measures, which include stain assessment, which uses the strictly standardized mean difference method to evaluate staining. There's a contingency table that evaluates the overall model performance and reduced chi-square values that evaluate modeled cell types. Alerts, flags, and equations can be set up to identify unexpected or out-of-range results. So all of these features and more actually combine to provide a comprehensive automated analysis that is interpretable, reliable, fast, and reproducible. So now I'm going to focus in on just a few of these features, and instead of telling you about this, I'm actually going to show you how and why this works. The driving force, as I mentioned earlier, behind GemStone is called probability state modeling. The original GemStone was released in 2009, and the paper describing the algorithm published here in 2015. So GemStone is not a black box. You can see your entire analysis, you can make changes, you can confirm for yourself that things are working as they should. There is no scripting or coding involved. So what is a model? A model is comprised of one or more cell types, such as CD4 or 8 positive T cells, B cells, etc. But these cell types are based on your panel and your experimental system. Within the cell types are expression profiles that control the fitting of the measurements in the panel. The expression profiles determine the probability of an event being included in a cell type. And they do this with what we call control definition points, which I'll actually explain shortly. So what is an expression profile? So an expression profile is actually a different way of looking at a 1P histogram. On the top right here is a very simple example of a DNA measurement with the typical intensity on the x-axis and frequency on the y-axis. If we take that histogram and turn it on its side, we now have frequency on the x-axis and intensity on the y-axis. To become an expression profile, we even out the events, so all events are distributed evenly over the expression profile. The x-axis becomes cumulative percent, which shows 0 to 100% of the events. The y-axis, again, is intensity. There is a peak finder system within the program that identifies the peaks based on the density of events. And you can see this little black triangle here on the right that shows you where that peak would be. We call this particular profile is called a constant profile. And there are some cell types that will only use constant profiles for enumeration. At this point in the modeling, this is when a raw data file is added, the events are actually in acquisition order, so you can see if there are any problems like clogs or bubbles during that acquisition. We can also model progressions. So if we try to do this with gating, we have the problem of subjectivity, double counting. We lose the ability to really look at those transitional events, and it's very difficult to scale up the analysis. The histogram shown here is CD27, which is expressed in this case in a peripheral blood B cell type. There's a negative peak here in blue and then a positive peak here in red. We can, to demonstrate modeling of a progression, we'll start again with that same CD27 histogram. If we turn that on its side, again, we have a negative population here in blue, the positive population in red. The x-axis is frequency and the y-axis is intensity. To become an expression profile, again, the events are distributed evenly along the expression profile. The x-axis is called cumulative percent. Progression here, it's percent of events. We can see the black triangles, which identify the negative and the positive population for CD27. So this particular sample is a B cell example, and we know that CD27 upregulates when B cells progress into memory B cells. We can see again that it's two black triangles which identify the negative and positive. The program actually positions the CDPs, the control definition points, based on those peaks for what we're going to call a step up, because CD27 upregulates. When modeled, the program actually rearranges the events so that the CD27 negative events are to the left here in blue, and the CD27 positive events are to the right here in red. We account for the overlapping transitional events between those two populations. The x-axis here shows us that approximately 80% of the events are CD27 negative, and approximately 27% of the events, 20% of the events are CD27 positive. The x-axis then becomes a common axis. If we look at this in an actual cell type, the common axis actually allows for scalability. In a cell type, the expression profiles are stacked, and each event will be evaluated and assigned a position. Every event in the sample file is accounted for. If I drew a line down the stack, we can see that the same event would be in the same position in each one of these expression profiles because all of these markers are correlated. This then becomes the basis of the overlay progression plot where you can visualize how the markers change and interact. In this case, it's B cells. We see CD27 here. It upregulates into the memory B cell population. These control definition points, for each expression profile, there's at least one set of these control definition points that define how that expression profile will fit the measurement data. So this is shown right here, and a little blow up right here for you. So each of these has 3 parameters that are associated with it. We have intensity, which is the Y position. We have a state, which is the X position. We have a standard deviation, which is also called line spread. It's showing us the 95% confidence limits. These CDPs are mathematical functions that actually define a probability distribution, and we will teach the model to select for that peak of interest by positioning the CDP on that peak of interest. The CDPs are flexible so that they account for variations in intensity, but they're constrained by what we call an estimation range to only select that peak of interest during the fitting process. And during the analysis, the program will calculate a probability for each event using all the information from all the cell types and all the expression profiles as well, and then calculate a total probability for the event, which will determine where the event is assigned. So again, every event in the data file is accounted for, and any unassigned or unclassified events are enumerated. So now I'm going to switch to GemStone and give you a demo of the wizards and the modeling process. So instead of telling you, I'm actually going to show you. And we will also take a quick look at the results and the analysis output. I'm going to bring up one of my GemStone programs. So when you open GemStone, you have a nice blank slate here to get started with. And if we wanted to create something called a project, we would click the New button, and we would be able to name our project. We could base it on a previous project that we collected. But I've also already set up a project, and I've just named this webinar demo. First thing I'm going to do is add my data files. When I click that Add button, it brings me to a navigation window where I can navigate directly to my project, or I can navigate out of that into a place where the data files might be stored. I'm going to open my folder here called Exported. And these are the same 5 files that we've been looking at. These have been previously cleaned with a model that I've built and done, and cleaned the data files automatically by removing the debris, removing the aggregates, and the dead cells. So I'll multiple select, I'll add them to my batch file. So we have 5 files now in our batch. To develop a model, I'm going to click this Develop button, which automatically opens up a tab that has all of the tools that I need to build a model. First thing I'm going to do is I will just select a file. I'll read in the file. And we'll know that the file is read in if we look here at the bottom right-hand corner, we see the name of the file, and we also see the number of events. The next thing I'm going to do, if I were going to build a model from scratch, I'd go to my measurement wizard and I would be able to look at all my different measurements. I would be able to adjust my scaling. I could edit the measurements if they weren't named correctly. I could also check my transforms and make sure that everything was on scale and transformed properly. But for the essence of time, I'm just going to skip right to my cell type wizard. I'm going to open that. And I have my different options in my cell type wizard. I can create a new cell type. I can create a report page. I could also edit the cell type if I had a template already built. Today, we're going to just create a very simple cell type. I'm going to create a CD4 positive T cell. I'm going to leave it as a competitive cell type, and I'll go to my next. Now, I'm going to talk a little bit about selection, and selection markers are really those iconic cell type lineage markers that you would want to choose for your B cells, maybe CD19. For T cells, we're going to do CD3 because we know T cells are CD3 positive. I'm going to just pick CD4 and CD8, very simple selection markers. I'm going to click next for my staging markers. We're going to talk about, this is really where you're going to define your cell type subsets. So if I'm looking at T cells, I might want to look at different subsets such as my naive, my central memory, my effector memory, or my effector subsets. I'm going to make this very simple today. I'm just going to choose CD27. I'm going to choose CCR7. I'll click next. And functional markers would be unknowns like cytokines or activation markers. I'm choosing just one for a simple example, and I'm going to choose PD1. And I'm going to go to next. So this is really where the fun starts, and we're going to actually build a model. Now we know that CD3 for T cells are positive. We see two distributions of events here on the right. We have two different peaks. So this would be the CD3 negative peak, and this would be the CD3 positive peak. Our CDPs come up on this positive peak. We see the line spread, 95% confidence limits. This is called the brightest peak. We also have some other choices here if we have different peaks that we want to identify. Right now, I'm going to leave it as it is because I want CD3 positive, and I'm going to hit preview. Preview actually goes through the data file, will identify the CD3 positive events and select those and fit that expression profile. So now we've actually selected all of the CD3 positive events in that sample file, and we can see right now there's 120,000 events. I'm going to click next, and which is CD4. Again, we want CD4 positive events. This is a normal distribution. I'll click preview, and now we've selected for CD3 positive, CD4 positive events. And you can see the number of events has changed to about 64,000. I'll go to my next profile, and this is CD8. This would be what we would call a negative selection marker. And what we want to do here is just sort of clean up the CD4 cell type by eliminating these dim to positive CD8 events. I'm going to change this to my dimmest peak. I'm going to do something called a low pass distribution. We want a low pass because it will grab all of the events from here below. So if we have any kind of unmixing or compensation issues, we're still selecting all those events that may be below 0 or maybe below the other negative events. I'll click preview, and then you can see it's cleaned that up. I'm going to go to our next profile. So here we'll do something a little different. For T cells, we know that CD27 downregulates as T cells become antigen experienced. So we call this in GemStone, we call this a step down. I have 2 peaks, and I have CDPs that are positioned on either one, on both of those different peaks. There's 2 sets that are linked. I'm going to shut off the width from peak, because what I want to do is I want to change that line spread to gather all of the, to consider all of those transitional events and to account for all the transitional events. And then I'll click preview. Right now the program is looking for all those CD3 positive events, CD4 positive events, CD8 negative events, and it's fit the expression profiles for CD27. It's rearranged the events so that the CD27 positive events are now to the left. The CD27 negative events are now to the right. And you can see this as a step down. If I go to my next profile, which is CCR7, you can actually see the data start to have some structure. It automatically will determine that you want to step down. I'm going to do this again. I'm going to just change that width. Okay. To 6. We count for those transitional events. I've got to go to next profile. Sorry, I should have just hit preview, but it actually I could probably go back, and I'm just going to show you. I don't know, maybe I'm not going to. I'll hit preview here. Okay, so we got back to the same place. So now all the CCR7 positive events are to the left, CCR7 negative events are to the right. And it's that difference between where CCR7 and CD27 downregulate that will form the staging. I'm going to go to my next profile. Here's one of my functional markers. I'll hit preview. This is what we call an unclassified marker. So what we're going to do with these is we're interested in these positive events for PD1. We will eventually put those into a tricom to enumerate those events, either singly or with other markers. So I click summary. I have a couple of choices here. I can save my template model, update the summary map, add to my Sensei, add database fields directly to the database, and I'll click finish. So you can see here that a report page has automatically been created. These are live. You can add any kind of dot plots. These dot plots are really here just for comfort so that you can see your data in a familiar way. We have some tables that are already created. This is a table of intensities, a table of statistics. Phenotype is here, and you can add more. You can add some, subsets or add additional pages. If we were going to build a model, a complete model, we would continue this process and go through each one of our cell types, adding CD8 T cells, B cells, monocytes, NKT cells, etc. We have a summary map which shows us what's been modeled. And then I'm going to come into my cell types. Each one of the cell types in our data file will have a workspace. The staging is calculated automatically. You can see that CD27 downregulates, CCR7 downregulates, and this again becomes the basis of that overlay plot. We can follow this over for CCR7 downregulation, CD27 downregulation, and the difference between these two form that staging. So for the sake of time, I'm going to just bring up another program. So this is a completed data file. I have done all of my subsets. You can see that there's a report set up with a summary table of statistics, QC table. I have my Sensei that's been calculated automatically. I have a report page for each one of my cell types. This is what a stain assessment page looked like. We have metrics that tell you what your stain, how well your staining actually is. I have a summary map with our unclassified events or unassigned events evaluated. I have all of my cell types. I have CD8, CD4, NKT, T star gamma deltas, B cells, NKs, etc., monos, MDCs, PDCs, and monos. I'm going to go back to my Sensei and just show you that this is actually interactive. I'm going to just bring up my edit window. I'm going to bring it over here to that. The populations here are automatically labeled to you. Again, this is calculated independently of the model, but it's linked into the model so that all of the populations are actually identified. This is, we can look at frequency, heat map, so you can look at where the populations are, and then we can look at intensity heat map, go through each one of our markers and scroll through, see what the expression is. The other thing that we can do is draw a little region around a population that might be unclassified or unlabeled. We can animate this. We can go back into the cell types and look where those events might be animated, and then we can actually scroll down through and look down at the phenotype for that. And it works both ways. So it's a process that is a two-way process. We could go into any cell type and do a lot of different animation. So the orientation of these sensays is the same. I'm going to bring up one more thing here. The project, everything is saved automatically in the project. It's there's a project library. A project consists of all these folders. I can come into my graphics. These have all been saved automatically. I can look at my different sensays. You can see that the orientation, the CD4 T cells are all oriented up here in the top right. And we can see this data file here that has a very different looking population. We also have PDFs that are saved out. We have different, all the graphics, a report. I'm going to come back here to my PowerPoint. Maybe. Okay, from this slide. Sorry, get a little bit of a lag here. Okay, I'm going to, this is the Sensei graph, which is looking at two of these donors. So we have this really pretty normal looking population here of CD4 cells, and then we have this other population that has kind of been brought out. So what happens when you have something unexpected like this? We can see that this is a very different population than the CD4s, but it is somewhat related. It appears to be related to that sort of terminal effector population, but it's separated by the CD4 T cell. We can look at this in a different way. Also, we can look at the overlay plots. We can see this greatly expanded effector memory or effector population. And we can see that CD57 is expressed here, CD56 is expressed, and PD1 is also expressed. You can look at heat map intensities to see the differences between data files. CD3, very bright for CD4, CD56 is expressed, CD57, and PD1. So in summary, I've shown you a number of different features in GemStone. These combine to give you a fast, automated, objective, interpretable, and reproducible analysis for any number of measurements or sample files. So importantly, GemStone also provides you a different way of looking at your data with these overlay plots, summary maps, Sensei, etc. So I don't have time to tell you more today. The project format makes collaborations very easy. You can share the whole project with your collaborators. Your analysis is secure and at your fingertips. And thank you very much. And thank you, Cytek, for the opportunity to talk today.
Thank you, Beth, for your presentation. I think we've got time for a couple questions before we transition to our last speaker. One is, what if you don't know the biology of all the different markers in the panel you're looking at, and you're trying to look for some novel subsets? What would you recommend for those types of users, those types of applications?
This question comes up a lot, and there is a reason that you develop your panel in the first place. So you have some markers that you are interested in. you have an idea about what some of the cell types would be. So, you know, depending on your experimental system and your either tissue, your blood samples, you have certain markers that you've chosen for your lineage markers, your sort of iconic markers, and then some of those unknowns. So what we really do in GemStone is we model what we know. So we know, you know, you really only have to model a few markers of what you know. And then it will very clearly show you what you don't know, and you have the opportunity to do that sort of exploration with GemStone. You have QC metrics that will help you. If you make a mistake, it becomes very obvious. We've done a lot of different cell types at a lot of different tissues at a lot of different samples, so we can always help you at Verity too. And, you know, we try not to encourage fishing experiments because I think, you know, most people don't, but but we know we all do that. So there are different ways to do it, and we do have ways that we can help you do that.
Thanks, Beth. I'm going to take one more and then we'll transition onward. Can you use the progression analysis to compare two samples? For example, if you have T cells that are treated with PBS versus T cells that have been treated with some kind of drug?
Absolutely. It's a really powerful way of looking at, because when we model things like leukemia, lymphoma with MRD, you know, those differences become extremely obvious. That the staging often is what changes. So it could be expanded. You might have a very expanded, if you're looking at T cells, a very expanded naive stage. If you're looking at, depending on what you're looking at, you can really see the differences between data files. Yes.
Great. Thank you, Beth.
You're welcome.
Before I introduce our next speaker, I want to launch one more poll for everyone out there. How often do you use R or code to analyze your flow data? I'll take a moment to select one of those, and we'll look at the results. Okay, looks like the result submissions are starting to slow down, so I'm going to go ahead and close out this poll. And here's what we've got. Looks like majority of people listening today do not use R code for their flow cytometry analysis, but there's definitely a good chunk out there that use it for some parts of it. Thank you for participating in that, everyone. Our next speaker is Elad. He is the Chief Executive Officer at Astrolabe Diagnostics and founder. He has over a decade of experience in computational biology, systems biology, data science in both academic and industry settings. Amir is an expert in the exploration of novel data sets, especially high-dimensional single event data, and in making machine learning and statistical analysis accessible to biologists and other colleagues. So with that, let me give the control over to you, Elad, and let's see what Astrolabe has to offer in analyzing this 33-color data set.
Thanks, Janelle. And I am patiently waiting for the presenter. While I'm waiting, I would like to thank Cytek for organizing today. And Janelle, thank you personally for all of the attention to detail and making sure this thing becomes reality. And I also would like to thank the crowd. I know that there are a lot of webinars going out there, and I appreciate the fact that you've taken a pretty nice chunk of your day to come and listen to all of us number crunchers talk about our different solutions. And
Sorry, Ayla, it looks like I keep toggling to you, and it keeps telling me it's waiting to view Beth instead of you.
We can listen to Beth again. That was pretty interesting.
Maybe if I toggle it away to someone entirely different. Let's see. Beth, is there anything popping up on your end at all that needs to be closed or acknowledged?
I have stopped showing my screen.
Oh, I got it.
Says
There you
waiting
go.
to view.
Okay.
Hey, hooray.
We just need.
Sorry about that, everyone.
No worries. So thanks again, Janelle. And today I'm going to tell you about the Astrolabe Cytometry platform for analyzing your flow cytometry data, and specifically the Cytek Aurora. And the motivation for Astrolabe is this idea that as your flow cytometry data becomes more complex, the analysis cost exponentially becomes exponentially higher. And to clarify, by cost, I mean any kind of resources that go into analyzing your data, whether it's time, whether it's the kind of bioinformatics expertise you need, and of course, money as well. And I imagine that most of the crowd here is intimately familiar with this area of the curve, where you have 10 or 12 markers, you're using something like the LSR2 or the Fortessa, and you can use traditional gating and straightforward statistics to analyze the different FCS files that you acquire. Now, we now know from experience with the Aurora and with other instruments, such as the Symphony and the Cytoph, that as the panels become more complex, as you're acquiring more samples, the analysis becomes exponentially more difficult. Gating becomes unfeasible. For example, any change in an early gate could really influence downstream gate. And also, you just need more of them. A typical data set might have 30 or 40 different gates, which is just too overwhelming for the average person. And furthermore, there's different statistical pitfalls that start to show up, stuff like multiple testing correction. And you really need to be aware of that as early as possible in your project. I think the worst case scenario is that you submit your paper, one of the reviewers is a statistician, and they shoot it down based on something you could have solved in the beginning. And I'm really appreciative of this day that Cytek organized. I think that out of the many instrument manufacturers that we work with, Cytek is the one which is most mindful of the kind of challenges their users are facing, whether it's building the panel, acquiring the data, quality control, and of course, analysis. And I'm proud to say that I have experience with the different tools that we've heard about today. And I used to think of these tools as the Swiss Army knife. The cool thing about OMIQ and FCS Express and GemStone is that they give you a wide suite of tools that you can use to apply to your data. So if you know what you want to achieve, and if you know how to get there, then these are fantastic tools, and I strongly recommend all of them. However, especially when new people come to the Aurora, especially when people are less experienced with bioinformatics, you're starting to face challenges. For example, you might not know the different algorithms, or you might not have the time to deal with them, and you're facing this flood of different methods and different tools, which one to choose, how to build the parameters. It can become quite overwhelming. Furthermore, if you've never used high complexity cytometry before, where do you start? What's the first thing that you need to do? And this is especially acute if you are running a core. Let's say you just got your first Cytek Aurora, you want to encourage your customers to use it, but how can you assure them about the analysis and the challenges that come with it? And I think in these kinds of situations, the Swiss Army knife starts to look like this. You look at the software, you look at the data, and what happens now? How can we deal with this? So the purpose of Astrolabe is to serve as a staging ground for your analytics. And the idea is that you acquire your data as always, whether it's human or mouse or non-human primates, you acquire it through the instrument, and then the single cell data goes into Astrolabe as is. You don't need to do anything. And the platform takes care of pre-processing the data, cleaning it, identifying cell subsets, and also fulfilling the role of the biostatistician by allowing you to identify cell populations or biomarkers that vary in a significant way across your experimental conditions. And again, the emphasis is that this is a turnkey solution. So you don't need to choose any algorithms, you don't need to set any parameters, you don't need to draw any gates. Astrolabe does it all for you. And when you start thinking about your experiment, you can think about the biology, you can think about the immunology, instead of having to worry about the raw data. So just to give you a brief idea of what Astrolabe looks like, I'm going to start a new experiment, and I'm going to call it Cytek Demo. And this is a cloud-based platform. We're following industry standards when it comes to data security, and we're actually working on Part 11 compliance right now. So you can click here to upload your files, and within a few seconds, we will have a button that is going to move us into pre-processing. I'm going to click on Define Features. This is where Astrolabe loads the files, make sure they work. And then we're facing this interface where you can define different sample features. And sample features are a way to explain your experiment design to the platform. Chris was showing something very similar in the context of OMIQ. So for example, we can add a sample feature called donor, and that will be the different donors that we have in this experiment. And of course, this is a bit like a spreadsheet. So if you have other relevant features, let's say you have different groups, let's say you have controls versus patient, you can add these as well. Or let's say you have different outcomes, such as responder and non-responder, and so on and so forth. All of these can be encoded here. So you do this, it takes about 10 minutes. Even if you have a huge experiment, even if you have 50 or 100 or 200 samples, and that's it, you're done. You click on continue, Astrolabe kicks in and starts going through the analysis. For these 5 samples, the analysis is going to take about 20 minutes. So instead of waiting, I'm actually going to fast forward to an experiment that I've analyzed earlier today. And I just uploaded this morning in preparation for today's seminar. So to clarify, to get from your FCS files to here, you do literally everything I've just shown you. Just upload the files and set up the experiment design. Now, Astrolabe is built around meaningful biological insights, and we want you to get as close to the biology as possible. Let me show you some of the ways in which the platform does that. And I'm going to start by switching to a view called the Cell Subset Navigator. This is a composite view of the entire experiment, and it's using data from all of the FCS files. In this case, we have 5 files, but even if you have 50 or 500, Astrolabe can analyze all of them. The biggest experiments we've analyzed so far were 350 million events, no downsampling, no concatenation, everything is done by the platform. So the starting point is the different cell subsets that we see here, and these are automatically identified by Astrolabe. So using your panel and using these series of different rules, Astrolabe can automatically label the different components, different populations in your experiment. And this logic follows traditional gating pretty well. So for example, to find T cells, we start by looking at CD3 versus several other markers, then CD4, CD8, and TCR gamma delta, and then markers for naive, memory, and Tregs. This can also be customized. So if you run your experiment, you see something is missing, we can add it for you. Then Astrolabe takes these different subsets and organizes them using dimensionality reduction algorithm. And what you see here is called the MDS map. And the idea is that this is a visualization trick to take the different subsets and put them on your computer screen. And the guiding principle here is that similar subsets are next to each other. So if I color code this by CD3, we will see that all the T cells are down here. If I color code this by CD20, then here are the B cells. And let's look at CD14. Here are intermediate and classical monocytes. So from the get-go, you can communicate with your data using the language of immunology. You don't have to worry about tailoring a model or about running t-SNE. You can start talking about naive CD8 T cells or memory B cells or so on. To me, one of the exciting things about these kinds of experiments is the kind of statistical questions you can ask. And in this case, we have 5 healthy donors. And we already know that there's quite a lot of variability in the immune system even within healthy individuals, how can we learn about that using Astrolabe? So one of the things that the platform allows you to do is to overlay statistical questions on top of the visualizations that you see here. So I'm going to use this tool to compare the different donors. And as soon as I do this, you will notice that the MDS map is going to be color-coded, and the color indicates the fold change across the donors. In other words, the deeper the color, the greater the variability between the 5 healthy individuals that we see here. And of course, if your experiment was responders versus non-responders, or if you have 4 or 5 different time points, you will see them here as well. And just by looking at this visualization, we will notice that there is one circle here which is bright red. And these are actually the NKT cells. If we click on them and scroll down, we can learn more about this specific subset. So what we're seeing here is a section which is dedicated just for the NKT cells. And first of all, we're seeing the frequency of these cells across the different individuals. If we had multiple samples per group, we will see aggregate statistics as well. Since this is one person per group, we're just seeing the bar charts here. And we will notice that one of the individuals, number 4395, seem to have a huge amount of NKT cells. And this is quite interesting. Going back to Beth's presentation, the Sensi, I believe that the Sensi map might have shown something similar. And here we can clearly immunophenotype these cells and learn that these are NKT cells. Furthermore, we can switch to the immunophenotyping view to see whether these NKT cells are different in this individual compared to the others. And what we're seeing here is a heat map where every row is one of the individuals, every column is one of the markers. One use of this is for validation. We can see that these cells are indeed CD3 positive and CD56 positive, but we can also see that for this specific individual, 4395, these cells are CD4 positive. They seem to be CD57 positive as well. And comparing to the bottom 3 individuals, they seem to be CD27 negative, CD28 negative. So we're already looking at biology. We're already thinking about what is going on with these 5 people. And we can see that one of them has a much higher population of NKT cells compared to the others. What else can we learn from this view? Well, Astrolabe is offering another visualization called the volcano plot. And in this visualization, every dot is one of the cell subsets. The higher it is, the more significant the trend for that subset. And you can read this from top to bottom. You can see that the NKT cells are the most significant trend. But another cell subset that might be relevant are these B cells over here. And once again, we can click on them and scroll down. And we will see that for most of the individuals, the naive B cells are somewhere around 0.2% of the population. However, for one of them, individual 4604, there's actually many more B cells than the others. Now, we might be curious, are these just the naive B cells, or are these the B cell population as a whole? So let's take a step back and try to look at all of the B cells instead of this resolution. So I mentioned that these different cell subsets are found by our labeling algorithm. We can also ask Astrolabe to collapse these. So I'm going to go to this area called compartment. And when I do, I'm just looking at the major compartments in the immune system. Specifically, I can look just at the B cells and scroll down. And when I do this, I will actually see that individual 4604 has less B cells than the other 4 individuals. So not only do they have more naive B cells than the others, they have less of the B cell compartment as a whole. Now, what about the other B cells? Well, let's go to the assignment level and let's look at the naive B cells, but let's also look at the 2 memory B cell compartments and scroll down. So for the naive B cells, we know this individual has more of them. For the non-switched memory B cells, this individual seems to have about an average compared to the other individuals. And for the switched memory B cells, they actually seem to have less. So once again, we're slowly fishing ideas and hypotheses about this experiment. One of them is individual 4395 has a lot of NKT cells. Another is individual 4604 has more naive B cells, and they seem to come at the cost of switch memory B cells. And all of these can be tied to other biological questions that you might have. So if we had clinical covariates about these people, such as gender, age, or disease state, we could have included this in the analysis. If we had, let's say, 3 different stimulations, we could have included these in the feature table, and all of the statistical analysis will cover these as well. So one last thing I want to look at is this idea of unsupervised clustering. And if you're going into the Aurora, one of the exciting things about the Aurora is that you can include these additional markers that have biological significance, but are not as straightforward to analyze, like, let's say, CD14 or CD20. And you might be familiar with methods such as FlowSum or SPADE or Phenograph that allow you to cluster the data. However, there's always a barrier to entry in using these methods. You need to understand them, you need to pick the right one, you need to choose the parameters, and so on. So Astrolabe eases the burden on that kind of approaches as well. And to illustrate this, I'm going to pick these same naive B cells as before. And if we scroll down, we see that they are much more common in individual 4604. Now I am going to switch to a different level called profiling. And when I do, you will notice that I have many more bubbles here. I have many more cell populations. And the reason is Astrolabe took each one of the canonical populations and further stratified them using unsupervised clustering. In other words, we start from what is known. We know that B cells and T cells are there. We know that T cells can be split into CD4 and CD8 and so on. Then we cluster within these compartments. So we've taken the naive B cells and we clustered them, and we end up with 5 different clusters. And we can use the Astrolabe visualization to quickly scan through them and see how they differ. Now remember, the trend that we've seen before is that the naive B cells as a group are most common in patient 4604. Let's see how do the clusters behave. So this cluster seems to be highest at 4604, as does this cluster. This cluster seems to be a bit different. And here is one cluster, which is about the same across the 4 individuals. And here is the last cluster, which is actually lowest in individual 4604. Now, this indicates that there is some heterogeneity within the naive B cell compartment. It's not that this individual has more of the B cell compartment, of the naive B cell compartment as a whole. It's just one group within them. And Astrolabe is actually giving us hints in the form of these markers that are in the title of the cluster names. Now, to clarify, this unsupervised clustering is using all of the markers. However, the platform is choosing some of them, which are most statistically different, just to guide your thinking and your exploration and your discovery process. So we're going to switch to the immunophenotyping view to try and understand how these cells are different in individual 4604 compared to all the other individuals. And one thing that is easy to notice is this presence of IgD. So we will see that it looks like these are IgD expressing cells as opposed to IgD negative cells, which appear equally across the different individuals. So once again, it's all about thinking about the data. It's all about building hypotheses. And in this case, we're looking at 5 healthy donors. However, we could have looked at disease states, treatments, and so on. So one more thing I want to show you is how Astrolabe can help you look at continuous markers. And one of the cool things about this panel is that it actually has markers such as PD-1, such as ICOS, chemokine receptors, and so on, which can be used to understand the behavior of the different cells here. And what I'm showing you here is called the differential expression analysis view. In this heat map, what we're seeing is all the different cell subsets as rows and all different markers as columns. And the colors, once again, indicate fold changes. The deeper the color, the greater the fold change for this marker and subset combination between the 5 different individuals. And we can very easily, for example, look at PD-1 and ICOS, and let's look at them across all of the T cells. So I'm going to scroll down and I'm going to switch to a feature box plot view. And this will reveal to us that individual 4604 seems to have lower PD-1 levels in the T cell compartment compared to the others. And that ICOS seems to be pretty variable across the 5 individuals. Then we can start drilling into specific subsets. So we saw the T cells as a whole. Let's compare the different memory and naive T cell compartments and scroll down again. And we can see that this trend where individual 4604 has a low expression of PD-1 is pretty consistent across all the different cell subsets. One possible exception is the effector memory CD8 T cells, where the first 3 individuals seem to be much more closer to that individual compared to 5036. Likewise, we can ask the same questions about ICOS, and there seems to be quite a lot of variability between the individuals and between the different cell subsets when it comes to this marker. Now, one last thing I want to touch on is this idea of quality control. I know that many people might feel queasy to use so much automation in their analysis, and it's important for me to clarify that everything that you see here is fully documented and fully transparent. As I mentioned, we're going through Part 11 compliance, so everything that we do must be accessible to the researcher who is looking at the experiment. And specifically, Astrolabe offers a reports export that has a pretty large amount of data in it. Just to show you an example of a few of the exports you can see here, we can see how Astrolabe cleaned the data. So again, this is all done automatically, and we can see how Astrolabe removed doublets and removed debris. We have one of these for each one of the samples, so we can make sure that we agree with the decisions that Astrolabe made. Furthermore, we can go into the labeling hierarchy itself. And one of the exports from the platform is this PDF, which we affectionately called Supplementary Figure 1. And this explains all of the analysis, all of the citations, plus biaxial plots. For example, if we click on T cells, Astrolabe is going to explain that the T cells are positive for CD3, negative for these other markers, and here's the biaxial plots. If we go to CD8 T cells, then again, it's going to explain the logic and the reasoning and the biaxial plots, CD45 RA negative, effector memory, and so on and so forth. So to summarize, Astrolabe serves as an entry point into your data. And I think that there is a lot of complexity inside the Aurora experiments, many different markers, experimental conditions, and so on. And Astrolabe can really help you tidy that up and get to the main ideas as quickly as possible. So now that we know that this individual has a high level of NKT cells, and this individual has something interesting with the naive B cell compartment, we can start designing our follow-up experiments. We can start sketching the figures for our paper. We can even start writing some ideas. So with this, I will once again thank you for your time. If you have any questions about Astrolabe, please feel free to reach out. I'm Elad at astrolabediagnostics.com, and I'm happy to take any questions that come from the audience.
Great, thank you, Elad, for your presentation. Let's get over to the questions. This one's got a comment at the beginning. It's very nice to have all the subsets labeled, but what if we are doing biomarker discovery and there's something we don't know the identity of? What do you recommend for those users?
That's a great question, and I would say that's a very common use case. And again, one of the exciting things about the Aurora is the flexibility to include these additional markers. And we have several tools that help you deal with that. The first one is the profiling level. And again, the profiling level includes unsupervised clustering. It's using all of the markers, including these markers that are not well defined, not well understood. And the same way that that we've used this mechanism to delineate the possible role of IgD in the naive B cells, this will be relevant for any other marker. The second tool is this differential expression analysis tool. And here you can explicitly state, here are the markers I find relevant. Here are the markers I'm curious about. Please tell me how they diverge across the different experimental conditions.
Great, thank you, Elad. Next one, is there a way to take into account absolute cell numbers into the analysis?
At the moment, we don't support absolute cell numbers. The mechanism by which existing users have integrated it is through this reports export. And one of the things you can download here are all of the cell frequencies and cell counts. And then you can plug in the equations for absolute cell counts there, whether using beads or any other mechanism, and transform the Astrolabe export into absolute counts on your side.
Okay. Next one is if you can talk a little more about where in Astrolabe the cleaning of the data and the pre-processing occurs, and also does the data need to have the compensation completed applied before coming to Astrolabe, or can you adjust that in Astrolabe as well?
So we do need you to compensate the data before you upload it to Astrolabe. Again, you're using the Cytek Aurora, so we need to unmix it. We need you to compensate it. Once that is done, everything else is done automatically by the platform. And the question was, can you show us where the cleaning is done? Well, as soon as you upload the FCS files, as soon as you start the analysis, the first thing Astrolabe is going to do is clean the data. And it cleans the data, honestly, based on what you know and what you expect. Going back to these figures I've shown you before, it's using forward scatter, it's using side scatter. If you want us to use CD45, we can use that. If you want us to use a live dead channel, we can use that. It's all customizable and the reports are available. We also have a quality control dashboard, which includes these statistics across the entire experiment. So you can quickly review the different samples. Again, you might have 5, you might have 50, you might have 500. This table is going to summarize all of the information and allow you to go through the cleaning results.
Okay, thank you, Elad. Next one, I'm going to read it directly to you. I'm a bit confused about the labeling. How is that done? Do you gate them manually prior to analysis? Do you tell the program to focus on some markers and then you call each phenotype whatever you want, like in t-SNE or FlowSum?
So there's no gating necessary on your end, and the platform works completely automatically. When you upload your FCS files, we read the different markers in the experiment from the FCS file, and we use this to tailor the labeling hierarchy. And the actual labeling is done by an algorithm called Ekbalam. We published it in June of last year, so it's publicly available. It's peer-reviewed. It's open source. And the way Ekbalam works is by clustering the data and then labeling it based on these rules. I will briefly mention that we've done several validation studies of this algorithm, including with Cytek themselves. And we see very high overlap between the labeling algorithm and manual gating.
Okay, great. Next question. How does Astrolabe platform tease out the batch effects of flow data?
That's a very common question. And I think if you're dealing with biology, whether it's flow cytometry or mass cytometry or any other assay, you should expect to see batch effects and you should be mindful of them. And we have several mechanisms in place to deal with these kinds of situations. First of all of this process of labeling and of clustering is done on a sample by sample basis. So even if you have some variability in the MFI of, let's say, CD3 across different samples, Astrolabe will correctly label the T cells in each one because, again, this is done on a sample by sample basis. Conceptually, this is similar to building a template in FlowJo and then moving that template around for each one of your FCS files. The second mechanism by which you can incorporate batch effects is you can explicitly analyze for them. You can include a column here called batch, and then all of the statistical analyses will be done over the different batches. And if there is something that differs between batches, you will know to eliminate it from the analysis of other experimental conditions. And finally, I will mention that as part of the differential expression analysis, one of the things you can indicate is a reference feature. So if you have reference spike-ins, or if you have a common sample that you acquire across your entire experiment, you can indicate that as the reference or the baseline, and everything will be corrected versus that reference. So even if you have, let's say, day-to-day batch effects, since they are reflected in the reference, Astrolabe will incorporate it into the analysis. So again, many different angles to attack this problem. We've seen some pretty extreme cases of batch effects, including situations where some samples were missing a marker completely, or some samples were fixed and some were fresh. And so far, the platform has dealt with them extremely well.
Okay, a couple more for you, Elad, and then we'll get back to some of the other questions that we missed from the earlier sessions before we wrap up. Is it possible to specify dim, medium, and bright populations within Astrolabe to help identify and phenotype rare subsets?
Yes, and by default, but the default labeling hierarchy doesn't do that. But if you choose to customize it, then we can work with you to identify different levels of expression of a given marker. Part of our service is really to discuss these kinds of questions with you. So if you are concerned about batch effects, if you are concerned about dim markers, these kinds of conversations are all part of our support mechanism.
Great. And this last one is, are there markers in Astrolabe, or markers that Astrolabe doesn't recognize or assign as a distinct population?
So left I checked the let's take BioLegend catalog as an example. The BioLegend catalog has, I think, over 400 markers in it. We don't cover all of them. Astrolabe by default covers, I would say, about 40 or 50 different cell populations, and these come from the literature review that we've done in-house, from resources such as the Human Immune Phenotyping Consortium. I would say they cover about 70 to 80% of the experiments that we analyze. For the remaining 20 to 30% of experiments, we work with the researcher to tailor these rules. One common example is if you have tissue-specific cells, for example, microglia or macrophages, we might not find them by default, but again, we can customize the platform just for your experiment, and it will find it. Again, this process is part of our service. There's no additional charge. It usually takes a few minutes, and we're done.
Great, thank you, Elad. Beth, I'd like to jump back to GemStone and cover a question that we didn't get to between yours and Elad's presentation. It is, where do you perform all the cleaning and pre-processing, for example, the compensation, scatter gating, viability, etc.? Where does that processing occur?
Yes, so in GemStone as well, the data does need to be compensated or unmixed. The cleanup, there's a couple different ways to do it, and I wish we had a little bit of time to talk about it, but the way that I cleaned this data is I built a separate model that looked at, I actually created a couple of equations looking at the ratios of forward scattered area to height and side scattered area to height. So I did it a little bit more algorithmically, and also looked at the live dead and created a model, a separate model that I could use on all of the data files. So it was a very consistent and way of cleaning the data. We have done that quite a bit with, particularly with higher dimensional data. You can create a cleanup cell type. You could also do the cleanup within cell types. The cleanup model works really well, the separate model. The data is automatically cleaned, and then the cleaned file is actually automatically exported, and you can add a, any kind of like a, if you noticed on the batch files that I added, there was a, they were appended with the cleaned word, actually, so I know that those are the cleaned files. So that it's set up in the batch and it can be done automatically. Does that answer the question? I think that answers the question. If there's another one, let me know. Yep.
Great, thank you, Beth. Sean, if I can ask you a couple more questions about FCS Express that we didn't get to. One is FCS Express 21 CFR Part 11 compliant? And the other one we missed is, can you do absolute counting and track that in FCS Express? Oops, do we still have Sean? Hopefully his audio's still there.
Sorry about that. I muted one button, but not the other. But yes, I'm here. Yeah, so great questions. So the copy of FCS Express that comes with the Aurora instrument is not 21 Part 11 compliant. but FCS Express 7 is ready for 21 Part 11 compliance. So what that means is if you want to add functionality to meet 21 Part 11 compliance for your lab, you can contact our team, and essentially we can add on to the software at additional cost what we call security and logging add-ons. And that enables your lab to help meet 21 Part 11 compliance through audit trails and security checks and things like that. So if you have additional questions, you can reach out to either me through my email or support@denovosoftware.com. We can give you more information about how to meet that compliance. And in terms of absolute cell counts, yes. So FCS Express has a tool called the concentration calculator in the software. And essentially that can read information from beads. It can read information from volume keywords like are required from the Aurora cytometer. And what will happen is all of your essentially gated populations will be reported out as absolute counts, and all of the calculations will be done for you in that report.
Okay, thank you, Sean. Elad, I have another one for you. For the algorithm that's used in Astrolabe, what if you disagree with some of the outputs that come from it? Can you override it at all and work with Astrolabe on what to consider for different subsets you're looking at?
Yeah, by all means, you can reach out to the support, to our support, and we will work with you to make sure you're happy with the results.
Great, thank you. And Diana, there were a couple more from your presentation that we didn't cover. One is, how should isotype controls be included and used in these large panels? Like for the 33 color one that we showed, do we have to make 33 isotypes and run those for this panel? Maybe Deanna's having the same double unmute difficulties as Sean.
Yes, I was. Sorry about that.
There we go. Welcome back, Diana.
I was saying that there are a couple of papers written by the rock stars in the cytometry field, Mario Roeder, Holdenmaker, and Rod Holzfast. And they have demonstrated that isotype controls are impractical and not recommended, especially for gating. And the thing is, antibodies bind specifically to your target through the FAD portion. And also they can bind specifically to an Fc receptor on the surface of some cells via the Fc portion. But the thing is, antibodies can also bind in a non-specific manner to low affinity targets via the FAD portion. Isotype controls were used years ago to identify non-specific binding in your antibodies. And the idea was to stain cells with an antibody that doesn't bind to your marker, but with the same isotypes, so the light and heavy chains, to chase non-specific binding. However, we don't know what the isotype antibody is binding to in the cells. The differences in the amino acid sequence in the variable regions of the new antibody might result in binding to any other epitopes in the cells. It's very difficult to make sure they have the same level of background staining. And also, you need to make sure that the fluorochrome antibody ratio is the same in both antibodies, that you use exactly the same antibody concentration, and even know that both antibodies have the same degree of aggregation. So if you're using multiple fluors in your panel, the best control for gating, if needed, are fluorescence -1 controls, in which all the antibodies are included except the ones that you want to define positivity for, or to evaluate issues with fluor spread. Other recommendation to prevent that non-desired staining would be using an FC block. To investigate also non-specific binding, isoclonic controls can be used where you add non-conjugated antibody to your reaction, and that will compete with your conjugate and then confirm that your fluorescence signal decreases when you add the isoclonic control.
Okay, thank you, Deanna.
But yeah, in general, they are not recommended anymore.
Got it. Got one more follow-up based on the isotype question. What do you recommend with respect to fluorescence -1 controls for these types of large panels?
So it's gonna depend on the resolution of your population, the expression levels, especially for markers that have dim expression or a continuum of expression where you cannot clearly see negatives and positives, and you need a gating strategy to define what's positive within your population. Then having that FMO in which all the antibodies are included except for the one you want to define that positive gate, that's where you would need it. So I guess depend on your panel, depend on the expression level of your markers. So let's say for checkpoints, case of PD1, TIM3, LAG3 markers that have dim expression, and it's very difficult to identify what's the positive population, then FMOs would be a good strategy for gating.
Okay, thank you, Deanna. So I think that
welcome.
Wraps everything up. I'd like to just thank all of our speakers one more time, Deanna, Chris, Sean, Beth, Ayla, great presentations. Thank you for taking your time to demo your software and show us the different tools that are available to all of our users out there today. This recording will be posted likely sometime this evening, maybe tomorrow at the latest, once it's done compiling and being processed. So we'll get that up on the Cytek YouTube channel, and I'm sure everyone else that's participating will have it on their websites as well. And anyone that is looking for contact information for the presenters, we'll shoot you an email with all that information tomorrow as well. So thank you again, everybody, and enjoy the rest of your day.